नहीं ये नहीं है किसी की ओ मैं एक मिनट understanding nano applicability. What we are going to explain here, the meaning of this lecture is that if you have the nanoparticle, how you could imagine that where you have to use them. So always think for some kind of useful applications where you could use them. Try to see in what way you could take the maximum advantage of the nanoparticle. So here I'll take you some of the environmental applications that how we can use the nanoparticles. It is not only the gold nanoparticle we'll be talking about, but you will have different kinds of nanoparticle. Here will be, you will see. So some other very important other kinds of silicon oxide nanoparticle, titanium oxide nanoparticle. So we'll be talking about their applications as well. Uh, This slide I showed you earlier. So here I mentioned that when we have to talk about the growth of the nanoparticle in the form of aggregated assembly, this is known as aggregated assembly. Schematically, we can show it like this. Uh, you are familiar with the nanoparticle when they are uh, present in the suspension individually placed. But when a group of nanoparticle is placed individually on a platform, that kind of assembly is known as aggregated assembly. So here what we are doing, in fact, we are growing the nanoparticles on micelle. You see, this is a very huge micelle. So you read the scale bar is 100 nanometer. Suppose if you asked me what is the size of STS, sodium dodecyl sulfate micelle, and usually the sodium dodecyl sulfate micelle is 1 to 2 nanometer maximum. So, but in comparison to sodium dodecyl sulfate, this is the micelle, which is a very big micelle, right? And the micelle, the formation of the micelle has taken place exactly the same way as in the case of sodium dodecyl sulfate. So now I'm going to tell you that this micelle is made up of polymeric molecules, not the monomeric molecules. When you have a polymeric molecule, it is a very big molecule. So therefore, polymeric molecule is expected to produce a very big micelle. So just for comparison, this is the SDS molecule. So when we have so many SDS molecule, they will aggregate in a micelle like this. So this is the micelle of SDS. Okay. But on the other hand, if I am taking a polymer like I think I have placed the picture, which polymer I am talking about, this one. If I take this polymer, you see now, this is this one is polyethylene oxide, CH2, ethylene, oxygen oxide, and you see the square bracket. It means that this part of the polymer is repeating how many times? 99 times. This one is polypropylene, polypropylene oxide, so it is 1, 2, and 3. Three carbon carbon functional groups, and this unit is replacing 69 times. So you could see that this entire molecule is in the form of a very long chain where these fragments are repeating 99 times, where the polypropylene oxide is repeating 69 times. Such kind of molecule or the polymer is known as block polymer. It means that you have one block of polyethylene oxide, another block of polypropylene oxide, another block of again polyethylene oxide, and schematically we can show it like this. It is polyethylene oxide, and then again you have a block of polypropylene oxide, PPO, and again it is oxide. Now the question is, this molecule forms the micelle, but what kind of micelle and what is the shape of the micelle and how the micelles are going to be arranged? So what we do actually, the polypropylene oxide is more non-polar. It is predominantly non-polar, whereas polyethylene oxide is predominantly polar. Not entirely polar, but predominantly. I'm using the word predominantly. So I can depict this this entire polymer in the form of a structure like this. So this is, you may say, PEO, PEO, and PPO, polypropylene oxide. When such molecules are polymeric molecules are going to aggregate, they are they aggregate in such a way that the predominantly non-polar part should be maximum away from the water molecule or the aqueous phase, where the predominantly polar part should be maximum closer to that of the aqueous phase. So how can I create a micelle for this? So it is like this. Sorry. So in this way, I can draw the micelle of uh, polyethylene, polypropylene, and the polypropylene units are directed in the center of the sphere of the micelle, whereas the polyethylene, they are away from, and this is now is the entire micelle. Now you can compare, this is the micelle of polymer, okay, and this is approximately, whereas this one is approximately 100 nanometer. And you could see this diagram, right? The black dots are in fact the gold nanoparticles. Now what happens because of the ether oxygen, so here you have ether oxygen in structure is lined with the ether oxygen like this, and there are lone pairs on the ether oxygens. Now these lone pairs can provide electron density to any electron deficient species, for example, Au plus 3. Au plus 3 is the gold salt and the gold ions, right? And what they can do, they can provide the electron density by providing 3 electrons. It can convert Au into atomic state. And once the atoms are created, the nucleation instantaneously starts, and then a nanoparticle is created. This is what shown over here. You see that, just a minute, let me see. Yeah. So this is the micelle which I have shown over here, right? So in this micelle, again, the way we explain that the conventional micelle is having the core and the stern layer, the perimeter. Also can differentiate the parts of the micelle, that detail in which we talk about for the conventional micelle, where I said that you have the core, inner palisade layer, outer palisade layer, and then the stern layer. But in this case, so the core, which is represented by the polypropylene oxide functional groups, those are embedded in the interior of the sphere, and whereas the polyethylene oxide functional groups, they lining the periphery. Now you see a cavity, the cavities are created as I mentioned over here. These are the cavities, these cavities. So the reaction is taking place, the nucleating centers from different gold nanoparticles like this. This is a tiny nucleating center. Nucleating center, if somebody asks you, the nucleating center is the is the first few entities, those when grow into full-fledged nanoparticle, and they usually possess few atoms, maybe 50 atoms, 100 atoms, or few hundred atoms. That is a nucleating center. The nucleating center itself is not a stable nanoparticle, but it is an entity which is in the course of the growth, and hence it will eventually converts into a bigger or full-fledged nanoparticle. So this nucleating center is created here in the cavity, and it grows into a full-fledged nanoparticle. It eventually grows into a full-fledged nanoparticle. That is why these black dots are the nanoparticle, and the black dots, all black dots are the gold nanoparticle, and they have grown from micelle. So exactly the way you might have heard about the template effect. So we have a template, and we are using a template in order to design a particular object. So exactly the same way what we are going to do, we are having the micelle. Micelle act as a template, and it helps to grow the nanoparticle, and the now nanoparticles are restricted to the surface of the micelle, and they will remain embedded in the micelle. This kind of assembly is known as a self-assembled state of the nanoparticles, right? So this is the transmission electron micrograph. If you want to see the scanning electron micrograph, this is the scanning electron micrograph. You see clearly the gold nanoparticle grown on the surface of a spherical micelle. Now, the mechanism for the growth is, I have shown over here. This is the surface cavity. The reaction which I have mentioned over here, electrons provided by the ether oxygens which are over here, ether oxygens. So these ether oxygens, they have electron density because of the lone pairs, right? And these ether oxygens are providing to the gold atoms. Once gold atoms are created, the nucleating center is created. That nucleating converts eventually into a full-fledged nanoparticle. So that nanoparticle grows all over the surface cavities. So this is known as a template effect. If somebody asks you, what is the template effect? Is the way that we could put the nanoparticle which is dispersed in the aqueous suspension independently. Can we direct them on a specified platform? You will say yes, it is possible. You could allow the reaction to take place on a platform or on a template so that all the nanoparticles which are going to be produced, they will remain on the platform that is aggregated state or self-assembled state of the nanoparticle. So we will talk about what is the advantage of this. So now, you see now nanoparticles over here, nanoparticles over here. So the advantage of this, uh, I'm just going to mention the catalytic applicability of the self-assembled nanoparticle in comparison to the nanoparticles those are dispersed in the solution. So here we have two reaction vessels. The one is where the nanoparticles are dispersed in the aqueous phase. So this is one where we have the self-assembled micelles, self-assembled nanoparticles on polymeric micelles like this. Now, which one we have to choose for a particular catalytic effect? It all depends on the reaction condition. If the reactant A plus B giving rise to C, and you want to facilitate this reaction, and you want that A and B should be facilitated to convert into C, and the gold nanoparticle act as a catalyst, and you will add A and B into the solution, expecting the reaction is going to be facilitated. If particles are going to catalyze the reaction, that's fine. But the thing is, if A and B, they are aqueous phase, solubilized in the aqueous phase, unable to approach the gold nanoparticle because of their solubilization properties, then you have to find out another way. How can you allow the A and B to see another site where they can undergo the catalytic reaction? I mean to say that if A and B are possessing some kind of amphiphilic properties, as I told you, amphiphilic properties where you have polar and a non-polar functional group, right? So then A and B, or only A or B, both may not have amphiphilic molecules, they would prefer the micelle. They will adsorb on the micelle because micelle itself is an entity which can solubilize, as I told you. That micelle has the ability to solubilize any kind of solute which is either the polar or the non-polar. Any range of polarity of the solute can be solubilized in the micelle. The polar will be adsorbed on the surface, non-polar will go into the core. So therefore, micelle is a unique kind of a reaction vessel or a sphere or a ball which can accommodate any kind of a molecule. So, these reactants will be accommodated in higher concentration in the micelle. Okay. And when they are in the micelle in higher concentration, gold nanoparticles are already there. The reaction which we are saying, A plus B giving rise to C, is much more facilitated. If I'm saying reaction vessel 2, reaction vessel 1 or 2, the reaction 2 is much more facilitated in comparison to reaction 1, because we have created a template which is not only the platform for the self-assembly of so many gold nanoparticles, but it also attracts the solubilization sites of the reactants whose reaction we want to catalyze. So therefore, individually, the gold particle effect may not be that enough, but they are in a large concentration over here, and they can facilitate the reaction to a very, very high degree of completion. So this is the main advantage of such kind of assemblies, and people are doing studying the several catalytic effect on the the self-assembled state of the gold nanoparticle on micellar assemblies. So this was the advantage of, I mean to say, for the templatic effect of the micelles in order to accommodate the gold nanoparticle and a really, really good substrate for any kind of catalytic effect. Now I'm going to take you to another kind of nanoparticle in self-assembled state. This was gold nanoparticle, right? Because we have learned about the reaction for in atomic state, starting the crystal growth, nucleation, eventually ending up with the formation of nanoparticle. Here we are going to talk about the silica spheres. What are the silica spheres? You see such a fine assembly of nanoparticle, right? Here, where is the scale actually? Scale is over here, right? So anyway, so scale is over here. So what is the size? Usually silica spheres have a size quite big, from nanometer to all the way up to 1 micron, but most of the time we are using silica spheres close to 200, 300 or 400 nanometer size. So you may assume that this is of that much size. What is the meaning of silica spheres? So what are they? They are in fact the silicon oxide. Their actual composition is silicon oxide and other derivatives with this. But how they are produced? There are few reactions which I have shown over here. This is the one, hydrolysis in the basic medium. So if you are going to take this molecule that is the derivative of silicon oxide, and you treat it in the aqueous phase in the presence of the basic medium, you can produce the hydroxide, right? And that hydroxide can be further used in a polymerization process, which is known as condensation, and eventually leading to the this other one is the other way is you can take tetraethyl orthosilicate ethyl. So it is CH2, CH3, CH2, CH3. There are four tetra, 1, 2, 3, 4. Tetraethyl orthosilicate. So it is the salt, it's a compound of silicon. So this is the name of this compound. When you are going to treat this compound in a medium of ethanol plus water facilitated by ammonia, and the condensation of the seeds are going to take place. So it is going to grow exactly the same way as we were talking about gold atoms. So it is base catalyzed hydrolysis, right? And slowly the nucleation sites, I mean to say that, so they are going in a network structure, uh, polymeric structure, to create a bigger and bigger sphere. So, and you see this is the first step, condensation of the silica seed, and that keeps on going till, till we continue the reaction. And eventually, we stop the reaction and we have silica sphere of the required dimension. If you want the smaller silica spheres, obviously you have to stop the reaction in a shorter period of time. But if you want bigger, you let the polymerization to happen, condensation to happen. Eventually, you have the bigger silica spheres. And you see they are means a controlled reaction condition can produce the silica sphere of very narrow shape variation, and they are all of the equal shape and size. Now, this is the way we actually create spheres. If you ask me where we are going to use it, most of the your sun screens, they are having the silica spheres, silicon oxide components, right? Apart from that, there are very important applications for these nanoparticles. That is the template effect. You see over here, template effect. What is the meaning for this? Now, suppose if I have uh, the micelles of SDS or CTAB, as I mentioned earlier, that you could have the micelles of CTAB or SDS with much smaller size in comparison to that of the polymeric micelles. Okay. Now, the thing is that how can we use them as template? Here we are using the micelles of conventional surfactant, SDS and CTAB as the template to create the sheets of silicon oxide. You see the sheets of silicon oxide with the pore size 1.5 to 10 nanometer. So this is the pore size. How can we prepare this? The condensation reaction which you have seen over here, if you are going to conduct on a template, you can create such assemblies, right? First, we are going to talk about this. In order to create this kind of silicon oxide structures, we are using the micelles as template. What you do, you have in the beaker you have the micelles, okay, the micelles over here, and the reaction which you are talking about over here, this reaction is conducted in the presence of the micelles, these micelles. So when the silica spheres will be created, they will adsorb on the surface of the micelle. The seeds are going to adsorb on the surface of the micelle, and if you keep on conducting the reaction, the seeds will merge and create a sheet. This is a sheet of silicon oxide. The way you have the silica spheres, the same way you have the silicon oxide molecules, condensation. The micelles, so this one is silica seeds. They are going to merge and create a shell. If you keep on growing this shell, now the shell is grown, right? And eventually you have another micelle over here and you have another shell, right? The shells are going to merge with one another, creating a three-dimensional structure, the one you see over there. This structure I'm talking about. And you see, these are the different micelles. So these different micelles, they merged together, created a template, and that template eventually created this kind of oxide, silicon oxide structure. Now, ask me that how, where are the micelles then? The micelles are here, and where are the micelles? These are the, because if we have to use the silicon oxide pores, empty for a certain application, we need to remove the micelles. What you do after the completion of the reaction, you take the sample, put it in the furnace, and heat it at the elevated temperature. Carbon is going to burn away, right, or decompose. So the micelles are going to decompose, and with the result of which you are left with simply this template of silicon oxide. This is the way we grow it. This is one way of creating the templated structure of silicon oxide, and we can use it for some purposes. So I'm going to talk about that as well. Let us assume that we want to create the hollow tubes of silicon oxide. These are the hollow tubes. You see, hollow tubes. It's completely, it's only that of silicon oxide uh, 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 kind of molecules, right? And they are empty. And this is the image. These are the tubes. They are hollow inside. But the question is, create. So they are created exactly the same way as we created over here, but instead of the micelles of the conventional surfactant, here we have to take the polymeric micelles, which are which have the shape transformation. Remember, I told you that micelles are going to transform their shape if the if we increase the concentration of the surfactant, and the micelle is going to take this shape. So there, so now here are the surfactant molecules, and this is the elongated micelle. So elongated micelle, right? And the shape transformation, this is sphere, and this is, you may say it's rod or a cylindrical micelle. If I take such cylindrical micelles and allow the reaction which I have conducted over here to happen, so I can create a sheet of silicon oxide, derivitized seeds over here. So now this is the same thing which we created over here. It is the same thing created. We have created on the spherical micelle, but here we have created on the cylindrical micelle, right? So it means that now the cylindrical micelle is the template. Here the spherical micelle was the template. Spherical micelle helped us to create this kind of structure with the empty pores inside, and the polymeric micelles, cylindrical micelles, created this kind of structure which is coated with the silicon oxide, and eventually when we treat in at elevated temperature, we will decompose the micelles, and hence we will produce the empty tubes. Now these empty tubes you could see here clearly in this image. This one is the scanning electron microscopic image, not transmission electron microscopic image. So these are the tubes. You see, so many tubes are here. So once you have created such kind of assemblies, it is up to you. There are plenty of applications for them. This can act as a filter for any kind of, usually the filter paper which you use, even the Waterman filter paper, very fine filter paper, the pore size is in micron. You never reach this pore size in nanometer, right? So therefore, there is no way you can have the filter paper with this this much dimensions. The reason is that this is the only way, means this can be achieved through the synthesis, not the mechanical process. There is no way you can convert small pores through the through any kind of I would say mechanical processing. It's not. It is through the chemical reactions only, because the chemical reactions involved atoms. So you control the growth or the reaction, eventually ending up to any kind of pore size or the morphology ranging from nanometer to micron or any other. If somebody asks you, what is the reason for that? Suppose I have a scissor, right, and I have to cut a piece, right? The scissor cannot cut a piece thinner than its blade. Am I correct? Right. If the scissor blade is already in micron, there is no way you can cut any piece smaller than the micron. The same kind of argument we can apply. So therefore, why chemists or the chemistry is that important for any kind of research related to the material science, because it is the chemist who knows how to use the different kinds of molecules or the self-assemblies of the micelles of different dimensions for the growth of any kind of other species. So I mean to say that when you start with the atoms and the molecules, eventually you can end up with the morphology with the pore size in the nanometer range. But if you are going to start with any kind of mechanical device which is already existing in the micron or bigger size, there is no way you can create a nanometer object. So this is the difference between the mechanical process and the synthetic process. If somebody asks you, why would you prefer the uh, 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 down-up approach rather than the top-down approach? Top-down approach is grinding the material to a smaller size, but bottom-up approach is starting with the atoms. So the thing is that starting with the atoms, atom is the smallest entity we have. Am I correct? Atom is the smallest building block we have. Any form of the matter which we have here, either in the solid state, liquid state, or the gaseous state, that is being created by the building blocks of atoms. So therefore we cannot slice the atom. There is no way. So any entity which cannot be bifurcated, broken up, so that is the smallest and the building block. So therefore when you start with the building block whose size is less than 1 nanometer, obviously you can achieve any object bigger than that. And that is the reason when you start making a room or a house, you start by using the bricks, because your ultimate aim is to create an object which is much bigger than a brick, right? So it's the same kind of philosophy we are going to apply for the bottom-up approach, because the bottom-up approach provides of numerous kind of possibilities to create a nanoparticle or nanomaterial of any dimensions, because we are starting with the atoms. So that is why here you have four size in the nanometer range, and is you have the tubes, silica tubes. So we are going to see that how can we use them. Okay, now I'm going to, as I told you that we have created this. So this was the template, right? And it's the same template which I mentioned in the previous slide over here. Template. I mean to say that if you keep, if, if, uh, if you take a high concentration of the micelles, you will have several interconnected templates like this. And this is the three-dimensional structure. You see, this template, silicon oxide, exists in this network structure. You see the pore size, right? This is the three-dimensional network structure. And the most surprising aspect of this is, read this. You take just 70 milligram of this sample, 70 milligrams. You could imagine that it's so tiny sample. Surface area it is possessing is equal to the one bedroom apartment. Surface area. The one bedroom apartment has plenty of open space. Suppose if you ask me how we can compare this, that is, it's not the size I'm talking about over here, right? So it is the empty space. There's plenty of empty space over here, but interconnected three-dimensional area is occupying that empty space and hence creating a much bigger surface area that is equivalent to this, right? What is the advantage of this? Now you see that, you know, the while drinking tea, you want to remove the tea leaves by using the sieves, right? And it is up to you that what is the mesh size, the bigger or the smaller. The smaller the mesh size, so even the small particles of the tea leaves, so they are going to be filtered out, right? So the same way, if you are having pore size much smaller, you want use this, you cleaning the drinking water. So this is the best kind of network structure in the nanometer range, with size pore size so tiny that it has a very high affinity to clean arsenic from the drinking water. Poisoning, the heavy metal poisoning, is highly toxic and it's cancerous, and therefore this kind of structure actually is has, apart from other application, I am just, because the easier way to understand that what kind of application we are going to use for this kind of material. So it is the cleaning of the contaminated water with arsenic. Apart from this, you can use it for catalysis, fuel cells, and the solar cells, because the silicon oxide and your solar cell composition most of the time, so they are the silicon oxide nanoparticle. So there are plenty of applications. So I mean to say that advantage of what I have shown in these few slides, I have just covered 6 slides. I have uh, I have mentioned that the advantage of the templating effect. If you have a clear templating effect, you know how to create the templates and what kind of assemblies you are going to use as templates in order to create the network structure, then there are plenty of opportunities in the material science. Okay, now we are heading another kind of bioapplication. You know the silica spheres. So these are the silica spheres. If you simply, for the time, yeah, over here, this is a scanning electron microscopic image. You see the fine silica spheres, clear spherical balls, 200 nanometer size, approximately 200 nanometer size dimension. The diameter is close to that, right? So this is the scanning electron microscope image, SEM, but this is the TEM. TEM can look through the object. So therefore, they look transparent because they are not metallic; they are oxide. So therefore, the diffraction is low, and hence they look semi-transparent. You see the boundaries over here clear, but the dark particles, from where you have a much more diffraction of the electrons, so those dark particles, nanoparticle, if I carry out the gold nanoparticle synthesis in the presence of these spheres, I can grow the gold nanoparticle on the surface of the silica spheres. What is the advantage? Again, I told you that there could be the advantage in the catalytic effect. So the silica sphere has the ability to absorb or to accommodate. It's a porous structure, and to absorb the reactant molecules. Once they are at the reactant, once they are in the silica spheres, then if the gold nanoparticle surface is the catalytic surface, and it can facilitate the catalytic reaction. Here, what they blood, you can use them in the presence of blood. So these are the silica spheres. Now you will be wondering that it is 200 nanometer, but here when we are going to place is to on the surface of the blood cell, they look so tiny, only tiny dots, because look at the scale bar. It is 2000 nanometer. Am I correct? 2 micron. 2 micron is 2000 nanometer. It is 10 times bigger than this. 200 nanometer, 2000 nanometer. So the blood cell size is much bigger that resides in the micron scale, micrometer scale. So therefore, there is a huge size disparity between the gold nanoparticle or the silica spheres and that of the blood cell. You can use them as a drug. Right. So here you see uptaking of the silica sphere by by the animal cell. You see, first the silica sphere in low resolution attached to the surface. Slowly it enters it is taken up and it is already inside the cell. Right? So in this way. Now, what is the meaning for this? The meaning for this is that if it is carrying drug or any kind of bioactive molecules which you want to use inside the cell assembly, right? And you want that it should be released inside the cell, so you need a carrier. You need a vehicle that can carry the load, a truckload of some amount of the substance, and that truckload of that truck will act as a vehicle that will carry it and enter into the assembly where it is going to deliver it. So that is the purpose of this. So there are many biological applications. So I would say that just look through the publications where you have applications of the silica sphere, and you will find many interesting applications probably on which you start your next research project. So how to become a scientist? This is a very important usual question. Many of my students are as well as the other students, so they ask me, I said, always start with a small thing. Just look in which direction you want to work and go through a publication, pick the idea, and see that how can you start some kind of preliminary experiment. And you start just preliminary experiment slowly, because among all the living organism, intelligent species is the human being. So God has provided you the brain. Eventually, you will see you have new ideas coming up. What if I'm going to take this reaction in this side, that side? That is the beginning of a good scientist. Anyway, going ahead, we are left with few minutes. So I have few more slides. Okay, so I have important slides because now I am taking you to directly the biological applications. Okay, this is BSA, bovine serum albumin, right? BSA, you are very familiar. This is the three-dimensional structure, domain 1, 2, 3. This is the globule, the BSA in its native state at 40 degrees centigrade, or we say room temperature. If you increase the temperature, the protein is going to unfold like this. This is unfolded from denatured protein, 80 degrees centigrade. Can we use the protein to produce biodegradable films? Now you know that we have strict rules and regulation not to use the plastic bags. And you will see that most of the bag, the written biodegradable bag, right? So is there any way that if a protein is freely available, I think you have plenty of proteins, gelatin is freely available, right? And in this way, you can use the protein to produce some kind of film that can be used for any purposes, for biodegradable purposes, for carrying bag, or for any kind of biological activity. So see over here, how can we do that? Gold ion in the presence of BSA at 80 degrees centigrade, it is possible to create a film. This film is created now, but you have to be very careful in arranging the mole ratio. When the mole ratio is less than 160, you cannot create the film. When the mole ratio is 167 or higher than this, then the film is created. This film is the protein film. But you see the dots created over here. The dots are in fact the gold nanoparticle. So dots are providing the reinforcement, the strength to the film, right? Otherwise the film is brittle and hence it may not be useful. But when you use the gold nanoparticle act as a binder. So hence the film is quite flexible and useful. Where is that? Just a minute. Okay. So what we do, we can choose BSA. Sorry about that. BSA is very expensive protein, you know. How can you use a BSA for this kind of purposes? But on the other hand, Zein protein is very cheap. It's a corn protein, actually, we we explained the zein protein. It's very cheap, freely available, because in North America we have plenty of corn. The way you have plenty of wheat and rice, but in North America when you drive across the cities from one place to, you see very big corn fields. So corn is the cheapest crop in North America, one of the cheapest crops in North America. And the zein protein is 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 a corn protein. So, but the problem is what insoluble. It's non-toxic, biodegradable, easy to process, highly brittle, and less flexible. And it is already frequently used because the non-toxic is used in few food packaging material, food packaging which is biodegradable, and chewing gums. The most of the chewing gums which you use every day. So zein is a part of that, and in pharmaceutical medicines. So it has something this kind of a structure. The protein, you don't need to fully understand that what is the meaning of this. It is the same way as we explain the BSA three-dimensional structure. The BSA three-dimensional structure was right. The same way the three-dimensional structure of zein protein is cylindrical structure. We call it cylindrical structure. Since it is highly hydrophobic, so the zein protein contains predominantly hydrophobic domains. So what we do actually, since it's not soluble in water, instead of dissolving in water because it's not possible, so what we take the solution. We take the surfactant solution, micellar solution. We dissolve zein, and in this way we prepare the zein solution. Now zein is dissolved in the micellar solution, producing a clear solution. What we do actually, we use the zein protein the way we use the BSA to create the film. We can also use the zein protein. So once you have created the aggregated assemblies of BSA and gold nanoparticle, to produce a big sheet of biodegradable film, we add the zein, because zein is much cheaper. We add the zein over here, and in this way we create a sheet. See yours? This is the sheet, and it is shown in the folded state by forceps. Right? So now this is made up of proteins only, but along with the proteins you have the gold nanoparticle, which help to maintain the strength and the flexibility. So where you can use it, so this is the tensile strength and strain at failure. So these are the experimental data. So how strong this system is. And, but for any kind of food packaging and other kind of a packaging where, so when this film is going to decompose, protein is instantaneously going to be decomposed or taken up by the insects or the available in the soil, and the gold nanoparticle, because the metallics, so they are going to be disposed of anywhere. So in this way, what I say that this is one of the best example of the protein films which can be used for any kind of purposes as biodegradable degradable films. So hence to solve the environmental problem. So here we have even cheaper. So you can use starch. Starch is really available. Right. So here we have prepared the films by using starch and glycerol. So starch and glycerol. So the glycerol coated cadmium sulfide. If you want to create a colored film, so here we have taken the cadmium sulfide nanoparticle, and here we have taken the gold nanoparticle. You see the color. The color of this film is slightly pink because of the gold nanoparticle. This one is yellow because of the cadmium sulfide. So cadmium sulfide electronic properties are much different from that of the gold nanoparticle. So it's not only I'm saying that it's not only as a packaging material, but apart from that, suppose if you want to study the electronic property in some other sense, then you can also perform that applications as well. So eventually, unfolding of proteins in the presence of nanoparticles can lead to the formation of structures, protein films, which, which have the wide applicability everywhere. Nanoparticles promote structural enforcement in biomaterials. So why we are using nanoparticles? Can't we use the simple proteins without the nanoparticles? The thing is, yes, you can use it, but the thing, by adding nanoparticle, it is providing the structural enforcement. So therefore, you may have the better applicability when you are using the protein films embedded with nanoparticles. Thank you so much for your attention. Any question? Just announce that the next. Good morning. We are starting with the next one. Application of iron oxide nanoparticle in blood cell extraction. So in this lecture, our focus will entirely be on the blood cell extraction. Uh, uh, the meaning for this is, suppose if you want to ask me that what is the meaning for this? Why we need to extract the blood cell? Uh, 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 in the in the in the testing of the blood, sometimes you need the blood cell separated from the plasma, and the conventional way of separating the blood cell is the centrifugation process. If you should centrifuge the whole blood, and you can separate the blood cell from that of plasma. We are going to provide a very easier solution rather than going through the centrifugation process. So the uh, 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 iron oxide nanoparticle provides a very viable solution to picking the blood cell from the whole blood, leaving behind the plasma. While performing the extraction, you have to be very careful that you are not supposed to break the blood cell, because when you are going to use the nanoparticle is very tiny in comparison to the blood cell. If the nanoparticle is going to interact with the blood cell membrane, they can break the blood cell and induce the hemolysis. So therefore, it means that we need a specific kind of iron oxide nanoparticles coated with some kind of biomolecules which have least affinity to interact with the blood cell to disrupt the blood cell membrane, but they can attach to the blood cell membrane without disrupting it, and in this way we can extract the blood cell. So this is the whole story on which this lecture is focused. We are using this same surface active magnetic nanoparticle which I here, the size close to 20 nanometer and coated with some kind of molecule. So we have two kinds, obviously, the surface active and water soluble magnetic nanoparticle. If you only look at this sketch, the first one, surface active magnetic nanoparticles are arranged at the surface, air aqueous interface surface, whereas the water solubles are dispersed throughout the bulk of the water. So what we can do, I'm going to place the surface active magnetic nanoparticle on the whole blood cell suspension, and likewise I place the blood cell suspension in the water soluble magnetic nanoparticle solution. And then what I see that if I'm going to, and for certain period of time, maybe 2 hours you can say, after that I'm going to attach, and I could see that when I attach the magnet, it's not only the magnetic nanoparticles are attached to the magnet, but magnetic nanoparticles are bringing the blood cells as well. So you could see that I have taken out the blood cells. And same can happen over here. You see the arrow. This arrow is going over here. This arrow is also going to this one. But then apart from that, we could see that there is an arrow which is coming over here and another arrow here. Apart from this sample, we have another sample which shows that this is one way of extracting it. While performing this extraction, you may break the blood cell and create a red color solution because of the oozing of the hemoglobin from the blood cell, and that creates the red color solution. Now, the amount of the red color solution is proportional to the of the blood cell broken. Amount of the red color solution that is created, that is proportional to the number of blood cells broken. So this is the whole story, and it is being tested with. I mean to say, we perform the UV analysis to figure out how many blood cells are being broken while performing this extraction. But the best magnetic nanoparticles are those which can extract the blood cells without taking. So we have to find out which are the best magnetic nanoparticles can be used in order to extract the blood. So this schematic representation is only showing you the experiments we performed with the surface active as well as the water soluble magnetic nanoparticles. Here is the extraction photos for the extraction of the surface active magnetic nanoparticle. This surface active magnetic nanoparticle soluble in hexane. We place the hexane suspended water surface active magnetic nanoparticle on the whole blood suspension. So this is blood suspension. We place them over here. Okay. Now, because hexane is not solid in the aqueous phase, hexane remains in the organic phase, and you have plenty of blood cells over here. The nanoparticles at the interface, they will start interacting with the blood cells. Now, after incubating it at 37 degrees centigrade for few hours. Place the, you see the magnet over here? We and and trap and extract all the magnetic nanoparticles. So those magnetic nanoparticles we extracted, we see that there is one possibility. If the blood cells are there, then when we are going to put it in pure water, this sample we are going to put it in pure water, blood cells are going to burst in pure water because pure water is not a saline, with not actually it is a buffer solution. So therefore, because of the osmosis, the blood cells are going to be burst, and you will have a colored solution because of the hemoglobin. The second one over here, the same concentration of the hemoglobin over here as that is over here. So what you can do, so you can make out that there is no extraction. So we can figure out by performing all these steps that if extraction has taken place or not. In this particular case, what we have observed that the hemoglobin is the hemoglobin absorbance is over here. This is the control, but this is the one from the sample. So practically there is no hemoglobin and there is no extraction of the blood cell. So all the blood cells are there in the solution, not picked up by the magnetic particle. But the magnetic nanoparticle has induced some sort of hemolysis. They actually break the cell membrane, and hence, in this case, when we are going to have the sample from this part of the solution, so we have a small absorbance. This one is a control. This one is a small absorbance. So it means that at the interface when the magnetic nanoparticles are placed, so we cannot have entirely facilitating extraction of the blood cell, and rather it is the magnetic nanoparticle involved in breaking the blood cell. So that is not our purpose. We don't want to break the blood cell. We have the other option that what we did here. We use the water-soluble magnetic nanoparticle. So here you see the water-soluble magnetic nanoparticle. And the water-soluble magnetic nanoparticle we picked and placed in the blood suspension. This is magnetic nanoparticle is the blood suspension. Now you might be wondering why it's not the red color. Because we have added magnetic nanoparticles, so that is why it's not red color. It's a dark color. And eventually when we are going to place the magnet here, we have seen that when we place the magnet and we take out the magnetic nanoparticle and we put it in the solution to see if there is any hemolysis. If there is no hemolysis, it means that no blood cells are broken. Right. So here we have taken that there is some degree of hemolysis. That's why it's a colored solution. Then the same experiment performed in another set of iron oxide. Now, what is the meaning of these two sets of experiments? In fact, they look identical. Whatsoever you see over here, the steps you see over here. Yeah. But what is different? Different is this experiments were performed by magnetic nanoparticles stabilized with zwitterionic surfactant DPS, TPS, HPS. Alanine is amino acid, glycine is amino acid, histidine is lysine and valine. So what is the meaning of this? The meaning is that we produce the magnetic nanoparticle by using a amphiphilic molecule as a colloid. As I told you that the magnetic nanoparticle can be stabilized with suppose if I say that this is a magnetic nanoparticle. Okay, so the magnetic nanoparticle is stabilized by a bilayer of amphiphilic molecules. So this is the monolayer, and then we have the bilayer. Now this amphiphilic molecule could be Gemini surfactant, conventional surfactant, amino acid. It could be amino acid as well. So these amino acids. But the amino acids usually they do not look like this because you do not find a much bigger non-polar functional group. But if you pick the non-polar amino acids, predominantly non-polar, you may have the same kind of amphiphilic properties as you could find with some other molecules. So in this way, amino acid molecule can also provide the colloidal stability, right? And in this way, we can use this as a, but it is not surfactant. This is water soluble. Water soluble. When we place such kind of nanoparticle in the blood suspension, and place the magnet over here, magnet, right? We want to see that how many blood cells are going to be extracted by the magnetic nanoparticle by using magnet. And the blood cells remain intact, they do not break. So here are the results. So we detect the breaking of the blood cell from the hemolysis. How much hemoglobin protein has oozed out in the solution? That is hemolysis. Hemolysis is the process by which the blood cells get break, and the hemoglobin which they contain, that hemoglobin is oozed out. So that process is known as hemolysis. In simple word, it's the breaking of the blood cell and producing the hemoglobin. So here you have the hemolysis and dialysis, right? What we observe that when we use all these amphiphilic molecules to create different kinds of magnetic nanoparticle, how many? 1, 2, 3, 4, 5, 6, 7, 8. We took different kinds of magnetic nanoparticle paired with different kind of amphiphilic molecules. It means that we have 8 samples of the magnetic nanoparticle. When we placed all 8 samples separately in the whole blood suspension, all 8 suspensions were able to break the blood cell. The breaking was up to 80%, close to more than 70% for the nanoparticles which were stabilized with glycine and alanine, and you could say close to 60% for histidine and valine, lysine and valine. Which is the least one? Least one are the zwitterionic surfactant. This is the zwitterionic surfactant. I have chosen only the abbreviated form of the name of the zwitterionic surfactant. Zwitterionic surfactant, why I took the zwitterionic surfactant? The reason is that the zwitterionic surfactant with the polar head group contains both amino as well as the acid group because there is zwitterionic and there is hydrocarbon tail. This is the zwitterionic surfactant. Now you see that ionic configuration chemically identically, it's identical to that of amino acids. So that was the reason to compare because the amino acids are also zwitterionic, right? So therefore, we took the amino acids, actual molecule, and the amino acid derivatized surfactant. We call this zwitterionic surfactant. We observe that if we choose to prepare the magnetic nanoparticle with only amino acids, they are highly toxic to the blood cell. This is very strange. Amino acid functionalized iron oxide nanoparticle. If I have a shell of only amino acid molecules, and now somebody asks you, this is very strange, amino acid molecule shell is going to break the blood cell. So suppose if you want to see that how it is happening at microscopic level, I say this is the cell. Okay, it's a disc shape. This is the blood membrane. It's a lipid membrane most of the time, right? Means it is predominantly containing lipid molecules. It's a cell wall, right? And if I have the magnetic nanoparticle coated with amino acid, and it means that amino acid has the ability to puncture or to break the cell wall. That is why the hemoglobin that is over here, so that will be oozed out to create the hemolysis. Am I correct? Now, so now we will try to understand that what is the chemical composition. Chemical composition of the blood cell membrane. I'll show it in the few slides after this one, but just schematically I can show you what is happening. The cell wall is in fact it's a kind of a structure where you have the head groups, because you have the lipid molecules, so they have the two hydrocarbon tails. So lipid molecules are creating the cell wall. This is the cell wall. Okay, cell wall. It's not only the lipid molecules. There are other molecules as well. You will see there are membrane proteins as well. Now, this is the head group of the lipid molecule. Most of the time it is zwitterionic or maybe anionic. Right? If I say that, okay, it is zwitterionic, and now I have the magnetic nanoparticle, and it is coated with a bilayer of amino acid. So amino acid is also like this, plus and minus. It means that I am initiating the electrostatic interactions between the positive end of the head group with the acid group, and the negative end of the head group with amino group, ammonium group. Right? So in this way, the electrostatic interactions will disturb the cell membrane, and hence it will break it, and leading to the oozing out of the hemoglobin. So this is happening in the case of amino acids, but it's not happening to that extent when we use, you see the blue color is that of the, yes, it's a C12. So when the docile, when this hydrogen tail is docile, then it is DPS. When it is C14, it is tetradecyl, it is TPS. When it is C16, it is hexadecyl HPS. So the effect is obviously less than half zwitterionic surfactant in comparison to the amino acid. It means that somehow the zwitterionic surfactant, they have less, though they are also breaking the cell wall, but not up to that extent. On the other hand, if I take cyclodextrin, do you familiar with the cyclodextrin? Cyclodextrin is it's a polysaccharide. It's a cyclic saccharide molecule. It's a ring-shaped structure, right? And if I take the derivatives of sugar, cyclodextrin, alpha, beta, dextrose, lactose, or sucrose, then there is no hemolysis except for this one, sucrose. They are not doing any breaking of the cell wall, right? And it means that the most of the blood cells are extracted. So here there is least extraction, maximum breaking. Here least breaking and maximum extraction, right? So Here, how many samples we use? 1, 2, 3, 4, 5. So we have 5 samples of sugar derivatized molecules which were providing the colloidal stability to the iron oxide nanoparticles. And what we observed that they are working excellently and they are extracting the blood cell without inducing the hemolysis. These are the experimental observations. So I'm taking you to the next slide. Now, where is the breaking of the blood cell? So these are the blood cells. You see the pictures? Where are the broken blood cells? See the puncturing, the holes in the blood cell, right? The holes in the blood cell showing that when the nanoparticle is interacting with the cell wall and it is breaking the cell wall, the hemoglobin is taken out, right? And now blood cell is broken. On the other hand, you see this blood cell, and you see these are magnetic nanoparticles. You see all these are the magnetic nanoparticles. Magnetic nanoparticles are attached to the cell wall. You won't expect just one, two, or few nanoparticle. There is plenty of nanoparticles. Those are going to attach to the blood cell membrane. So here you have so many blood cells, all covered with the magnetic nanoparticles. So here you also have, so you could see here, it's not clear because it's a 5 micron. You see the range? It's a very big range. But here it is 500 nanometers, so that is why you could see nanoparticle. When the nanoparticle size is too tiny, only it is 20 or 30 nanometer, whereas the blood cell is micron, 5 to 6 micron in size. So there's a huge disparity between the size of the blood cell and magnetic nanoparticle. So that is why you may not see clearly, but only you could see that all these are the magnetic nanoparticles. We can perform the matching and you see proof. So this area is actually covered by the iron oxide nanoparticle. This is the mapping for the iron. This is the mapping for the oxygen to confirm that indeed all the particles which are present, they are made up of iron oxide. Okay, so this is the way we see. This slide is extraction and blood cell morphology. So humanoid cell. So here we have shown you these particular two images. So they are showing you the humanized broken blood cells. But you would like to see the unbroken blood cells. See over here. You see clearly the blood cell? The disc-shaped blood cell? These cells are not disc-shaped because their morphology somehow altered. It means that all blood cells in this sample, they were interacting with the magnetic nanoparticle, but in most of the cases, the cell membrane got punctured or disturbed, broken, and some cases, the morphology of the blood cell from the disc shape has been changed. It's not a disc shape. That what is a normal blood cell shape is this one. You see the bowl shape, disc shape, it is the disc shape blood cell. Now, you see the disc shape, all these blood cells are intact. They are in its natural shape and structure. No alteration in the structure of these cells. So, extraction and blood cell morphology. So here, non-humanized cell. So this extraction is the best extraction where the morphology of the blood cell has not been altered. So therefore, if you ask me that which were the successful experiment, these were the successful experiment. So it means that the images where you see the intact blood cell, those were images of these samples. So these samples where no hemolysis has taken place. You see over here, this color is why this color. Most of the blood cells are extracted over here, right? When you put this sample in pure water, the blood cells got burst and created the blood. Right? On the other hand, this one and put it in pure water, there is no color. What's the meaning for this? There are no left blood cells in this sample. That's why there is no color. Here, all blood cells are extracted intact. So when I put them in pure water, they are going to burst. So the intensity of the color is corresponding to the number of blood cells extracted. As more and more color is there, more and more blood cells are extracted. And that's what you have seen in the image. Then, what is the conclusion for these experiments? How can we use this technique to replace the centrifugation method? What we should do? We should come to the mechanism. Here is the mechanism. So I have divided this into a complicated image. I'm going to explain it. Okay, first I'm going to show this is the morphology, molecular arrangement of the blood cell membrane. This is lipid bilayer. This is the surface of the cell. Outside is water. Right, outside is the aqueous phase. Here, inside is the blood cell. So I'm showing this the surface of the blood cell, and the membrane of the blood cell is mainly composed of that is a lipid bilayer. See the structure of the molecule. These are the lipid bilayers. Okay, in this lipid bilayers, actually I have not labeled many other things here. So this one is the membrane protein. You know the membrane protein that provides the reinforcement to the cell wall structure. That is something like this. Okay, so then there are other components also. Okay, I should have labeled them, but anyway, apart from this, you have the glycolipid molecules. These are the glycolipid molecules. Okay, and these are the sugar derivatives, sugar molecules, and below the cell membranes, immediately below the cell membranes, you have the spectrum. It is the arrangement of the membrane skeletal structure. It provides the support to the membrane of the blood cell. Okay, what happens now? Well, I have divided. Initially, you concentrate on the cell wall and the spectrum, but actually the cell wall is in action rather than any kind of involvement of spectrum. So it is the lipid bilayer membrane, so that is in action on it, interacting with the magnetic nanoparticles. I have divided the diagram into two parts. This part belongs to the hemolyzed where the hemolysis has taken place. Hemolysis has taken place, it means the membrane has broken down and the protein has been restored. Right. This part belongs to where no hemolysis has taken place. See over here. Hemolyzed blood cell, non-hemolyzed blood cell. Now, microscopically, what happens? See over here, when I am going to take the amino acid stabilized magnetic nanoparticle, they are stabilized like this. I'm using the same kind of a picture over here, the bilayer or the coating of the amino acid. When they come in close, in contact with the cell membrane, these are the head groups of the phospholipids. It's the same kind of interaction which I showed over here. The head group of the phospholipid molecule, and this is the dipolar amino acid. So the electrostatic interactions are taking place, with the result of which this membrane is disrupted and hemolysis is set in. The protein is oozed out, and therefore these nanoparticles which are coated with the protein, or these are the biological surfactants. Even biological surfactants are not working. They also have the ability. They have the ability to break the cell membrane, and we can use for the extraction of blood cell. So it means that these magnetic nanoparticles, which are produced with various kinds of amino acids and the biological surfactant, They are not good magnetic nanoparticles for extracting the blood cells. But what are the good? Cyclodextrin. Cyclodextrin is, let me just show you the cyclodextrin is kind of a structure like this. It's created by the aromatic rings of the sugar, and there's an empty space over here. It is non-polar from inside, polar from the surface because of the hydroxyl groups. Right, that is the cyclodextrin. I have shown this cyclodextrin in the bucket-shaped molecule. You see the bucket? The bucket-shaped molecule. The bucket-shaped molecule is arranged on the surface of a nanoparticle. Right, and they are providing the colloidal stability. They are amphiphilic molecules, so therefore they can be used as a colloidal stabilizer. What is happening? This bucket-shaped molecule has a cavity inside. The glycoprotein, which has the sugar and dieting, that can be incorporated into the bucket. This cavity inside the bucket, it accommodates the glycoprotein, sugar and dieting, and because of the non-polar interaction. So therefore, magnetic nanoparticle is not coming into the vicinity of the bilayer membrane. It is entangled over here. Magnetic nanoparticles are entangled over here and are directly interacting with the bilayer membrane. They have also the ability to interact with the bilayer, but most of the magnetic nanoparticles do not interact with the blood cell membrane, though I have shown few of them. We cannot entirely exclude the possibility of interacting with the lipid bilayer, but most of the magnetic nanoparticles, they interact with the glycoproteins, with the result of which the membrane, bilayer membrane, remains undisturbed, and hence the anchoring of the magnetic nanoparticle. There are several magnetic nanoparticles. Those are going to anchor. You see, several magnetic nanoparticles anchoring to just one cell, and hence when you are going to apply the magnet, placing the magnet is attracting the magnetic nanoparticle because blood cell is not magnetized, right? So then you are applying the magnet, the whole is dragged towards the magnet. This is the extraction, the whole assembly. The meaning of this is, now this assembly contains not only magnetic nanoparticle, but also the blood cells. So when we are going to place the magnet over here, so a magnetic nanoparticle along with the blood cell, they are being extracted, and that is why they are over here. So in this way, what is the mechanism? The mechanism is suggesting us, when you take the magnetic nanoparticle stabilized with sugar derivatives means which one? Cyclodextrin, alpha, beta, dextrose, lactose, may not be, sucrose is not good. So if you're going to stabilize with these sugar derivatives, then they are very good carriers of blood cell. They are very protectors. They are to break the blood cell, and then you can use them for extracting the blood cell, leaving behind the serum, and without performing the centrifugation, you can simply take out the blood cell. And this is a new technology, new methodology, and you do not need any kind of mechanical centrifugation process to separate the blood cell. It's a very quick, simply put the blood cell, magnetic nanoparticle stabilized with cyclodextrin or the sugar in the whole blood cell, place the magnet, take it out, and you have the blood cells. So this is the meaning of the whole story. Okay, here is the summarization. So summarized all different kinds magnetic nanoparticles we use. Extraction versus hemolysis. We want least hemolysis, maximum extraction. That is the meaning of the usefulness of the methodology we are producing. We want that least hemolysis being induced, and we want the little number of blood cells are broken, but most of the blood cells are extracted. Okay, so we have prepared all the different samples of blood cell. You see, 0 to 100% is the hemolysis. So in this case, this is the structure of cyclodextrin. As I told you, the ring-shaped structure. This is the cavity equated with the bucket-shaped structure. It is basically a bucket-shaped molecule and empty from inside. It's mainly non-polar from outside, right? So when we use dextrin, we also call oligosaccharides, means sugar molecules arranged in a cyclic arrangement. That is the meaning of oligosaccharides. So when we use such kind of molecule for the synthesis of the magnetic nanoparticle, there is the least hemolysis, maximum extraction. More than 50% of the extraction, right? Take dextrose. For that, effect is almost the same. Dextrose is also useful as much as cyclodextrin. Biological surfactant, sodium cholate. This is sodium cholate. If you look at the internet, it's a cholesterol derivative, biological surfactant, right? If you take sodium cholate, they're giving us any better results. So there is still higher hemolysis in comparison to this, right? But the extraction is much less, 50% than this. To zwitterionic surfactant. This is zwitterionic surfactant, amino acid. If you take surface active, if you take the surface active, all those are from here to here, they are the water soluble magnetic nanoparticle. Surface active magnetic nanoparticles are not working. So in the case of surface active magnetic nanoparticle, hemolysis is too much, extraction is very less. So this is the conclusion. And if you ask me that among all magnetic nanoparticles, which are the best performer, I would say that they are the sugar derivatized, stabilized magnetic nanoparticles are the best one to extract the blood cell. So, you may ask me that when next time you go for the blood test, would you expect that the people in the blood lab using this kind of technology? I'm not sure, at least immediately. Maybe after some time, probably they may use such kind of technology rather than centrifugation your blood sample to extract the blood cell and to perform the test and to test the serum and for any kind of other purposes. So, but this is a new kind of a technique, methodology can be easily used and is much easier to be used in comparison to the centrifugation. Conclusion, use surface active magnetic nanoparticle with less potential to disrupt blood cell membrane. Now, the surface active magnetic nanoparticle I have shown you, they are not good enough. They are not good enough for the extraction, but we need more experiments. Probably we may have some other kinds of surface active magnetic nanoparticle. They can induce less hemolysis and may prove better extractors, and they can work exactly the way we have shown you water soluble molecule. But for the time being, not working. So they have the less potential to disturb blood cell membrane biological applications at biointerfaces. Now, it has several implications. I mean to say that when you are using the iron oxide nanoparticle for hyperthermia, magnetic hypo, MRI, tissue engineering, you need the magnetic nanoparticle which do not disrupt the blood cell. So therefore, when you have to perform those kinds of experiments, you would prefer to use the magnetic nanoparticle which are not breaking the blood cells. So that is the meaning for that. Cyclodextrin, dextrose, stabilized water-soluble magnetic nanoparticle are the best components of the water-soluble magnetic nanoparticle for bulk biological system or the extraction of the blood cell from the whole blood. So this is all about regarding blood cell direct application on which this topic was iron oxide nanoparticle for biomedical application, right? But you could see that in order to arrive at this concluding topic, I had to walk through so many lectures, right? If I could have directly come to this lecture, you could imagine now you have gone through all the lectures, how difficult it was for you to understand what is happening. So that is the one when we design the course, we always design the course. We start with the basic and slowly we go ahead to understand the complicated concepts. So that is the best way of understanding the science, right? Science is not a story where somebody is telling you the story and it's directly going into your brain. It is the evidence which you have to prove. It is the quantification which you have to measure. The evidence has to be quantified. So somebody can say that it's a weak evidence, strong evidence. It's not that way the science is being explained. Evidence means explained in terms of the numbers. We say this is the evidence, these are the numbers. To prove that is a strong evidence, that is known as the quantification. So we are left with just one lecture in the afternoon. So we will see you again. So thank you so much for your kind attention. You can have any question now. Thank you. Yes, please.
Has iron containing proteins. So, sir, why we can't use that things for the magnetic field? Why we put that nanoparticles?
You are asking, you are saying that the blood proteins containing iron.
Yes, sir.
Can, can you repeat your question once again?
Sir, blood proteins
Yeah,
contain iron proteins. Like
correct.
so, sir, why means till now we use centrifugation for extractions, yes. But then why we need nanoparticles?
Yeah. So that's a very valid question. So you know that the blood protein is a hemoglobin, and the heme group you already know. It's the in the center of the heme group there is iron that is coordinated, right? So she's talking about that iron. Now that iron is not magnetized. It cannot interact with the external magnet or the magnetic field. So here the purpose of the magnetic nanoparticle is to attach the magnetic nanoparticle to the blood cell. We can use any other nanoparticle. Suppose if you ask me, why can't we use the gold nanoparticle, silver nanoparticle for this kind of extraction? I would say that the silver or the gold nanoparticle may not be having that much advantage as much the iron oxide. The reason is they are not going to be attracted to the magnet. So here we are talking about the ease of the extraction. The ease is when the iron oxide nanoparticles attach to the blood cell, when we place the magnet, the whole assembly is attracted toward the magnet. If I am using gold nanoparticle instead of iron oxide, I may place the magnet, they are not going to be extracted. Again, I have to use the centrifugation method. So in order to avoid the centrifugation method, iron oxide nanoparticles are used. They are attached to the cell wall. They are not breaking the blood cell, but they are attached. So therefore, when I apply the magnetic field, magnetic nanoparticles are bound to be attracted towards the magnetic field, but they are attached to the blood cell. They are dragging the blood cell towards the magnet. In this way, we are extracting them. So that is why it's the ease with which. So in when you have to use the application or you use the magnetic or any other nanoparticle for any kind of application, it is the applicability, it is the ease with which we are going to apply it so that the methodology which we are creating, that is much better methodology economically, time purposes, and with the ease of achieving the objectives of that methodology in comparison to the same methodology already existing. So extracting filter means, if somebody asks you that in what way this technique is better, you'll be saying that we need to extract the blood cell from the serum to perform certain tests to see if you have anemia, right? And there are other things you have to see if other critical illness is related to the shape of the blood cells, right? So for all those purposes, if you have to remove the blood cell from the whole blood, leaving behind the serum, you will be performing time consuming centrifugation technique that is energy intensive as well as time consuming. But this one is not energy intensive. Simply picking the magnet, attaching, blood cells are separated out, and you can take out the blood cell and see through the microscope if their shape or the size is the same. Otherwise, you have the anemia. That is the
Active nanoparticle, it's only depend upon the surfactant molecules.
correct. Absolutely. It is the Gemini surfactant. It is not only one kind of Gemini, it is a whole class of molecules and several series of Gemini surfactant. It is your experience performing the experiment will teach you that which kind of Gemini surfactant is most suited to create the surface active magnetic nanoparticle. Not all Gemini surfactant, and just few of the conventional surfactant can lead to the surface active magnetic nanoparticle. But most of the Gemini surfactant can help you create the surface active magnetic nanoparticle. Is it okay? Any other question, please? Yes, please. Excuse me, please. Once again.
There was discussion about the incorporation of nanoparticles in films.
In films. Okay. Yeah. So you are asking me?
How does the incorporation of nanoparticles enhance their structural stability?
Yeah. Okay. Just a minute. Actually, there was another diagram which I've not included in the slide, so I'm going to draw it now. This is magnetic nanoparticle. This is magnetic nanoparticle. Okay, the protein is something like this. Okay, and you have another protein that is something like this. So the proteins are adsorbed on the surface of the magnetic nanoparticle through hydrophilic domains. Predominantly, hydrophilic domains have to adsorb on the transition metal nanoparticle. So you may expect that this is the hydrophilic domain that is on the surface, acting as anchoring site to attach the protein to the nanoparticle. The same is the story over here. But where are the hydrophobic domains? Hydrophobic domains are away from the surface, and because of the known polarity, the hydrophobic domains of this protein molecule, they have the non-polar interactions. These non-polar hydrophobic interactions through the hydrophobic domains of adjoining protein molecules, they are very intense, right? So therefore, you see that this protein macromolecule is attached to this because of the nanoparticle. Helps to bind the protein molecules and to create a reinforcement in the form of a protein film. Okay. Any other question?
Thank you so much, sir. Now there will be a short tea break for 15 minutes, after which there will be tutorial conducted by Dr. Rajiv Jindal. Here only we will have the PPT.
So, good morning to all of you. Good morning. So, we are in the last tutorial number 5, that is applicability of surface active iron oxide nanoparticles in removing metal nanopollutants from aqueous bulk. So, these are the contents of the presentation. Now, in the previous experiments, we have done 4 experiments so far. Now, the present experiment is related to experiment number 3 and 4. 3 was synthesis, characterization, and electronic properties of gold and silver nanoparticles. And yesterday's practical was synthesis of Gemini surfactant stabilized surface active iron oxide nanoparticles. So we will be using these two, the third experiment and the fourth experiment, to extract the silver and gold nanoparticles with the help of iron oxide nanoparticles. So the metal pollutants, any metallic chemical element that is toxic at low concentration and has a relatively high density. They can be used by a variety of environmental activities, including metal mining, steel production, metal leaching from landfills, manure, and water runoff, among others. Rapid industrial growth is the primary driver of metal contaminants. Gold and silver metal nanopollutants can also pose greater threat to aquatic life when are leached into the water resources. Next. So these are the harmful effects of metal pollutants in general. So what are the harmful effects? The toxicity, chronic exposure, environmental impact, neurological effects, respiratory system damage, carcinogenic potential. So silver is a metal pollutant. So silver is toxic heavy metal. It affects environmental and human health. Then it causes argia, lungs and throat irritation. This argia is the skin disease that is caused by silver. Then the sources of the silver pollutant are industrial effluents, medical waste, agricultural runoff. Similarly, gold. Gold is chemically inert and stable metal. Long exposure causes ingestion, allergic reactions, etc. Gold nanoparticles can damage DNA cell. So the sources are industrial activities, gold mining, and urban runoff. So in today's experiment, we shall carry out the extraction of silver and gold nanoparticles. So in the third experiment, we have synthesized iron coated with SDS. So this is the iron coated with SDS, and this is the silver with SDS, and this is after titration. We have taken the magnetic nanoparticles, we have mixed these two. At the interface, there will be the extraction of gold and silver nanoparticles. So how this is to be detected? How the extraction of gold is to be detected with the help of UV visible spectroscopy. Now we take the blank sample. Blank sample means without any magnetic nanoparticle. So it will be having high concentration of the gold nanoparticles. This is for gold. It will be having the high concentration of gold nanoparticles. Now, if we go on increasing the concentration of magnetic nanoparticles, what will happen? As we add magnetic nanoparticles, the more and more gold nanoparticles will be extracted. Okay, so its concentration will increase or decrease. If it is getting extracted, so in the solution it will decrease. So this is how the concentration goes on decreasing, right? 13.33, it is at the bottom. The concentration goes on decreasing with the help of UV visible spectroscopy. Next, similarly for silver, it also goes on decreasing at 11.42. It is the minimum, right? So we will be carrying it out with the help of UV visible spectroscopy. We will monitor the reaction with the help of that. Next, metallic nanoparticle extraction. The extraction of metallic nanoparticles was carried out by mixing 1 milliliter of an aqueous suspension containing SDS stabilized gold or silver nanoparticles with 1 ml of hexane soluble surface active magnetic nanoparticle suspensions in a screw capped glass wire. Okay. The magnetic nanoparticles that we synthesized yesterday, it was soluble in hexane. So it is taken in a vial this is soluble in hexane. Then this resulted in the formation of a biphasic system with equal volumes of aqueous and organic layers. The aqueous phase appeared pink for gold and yellow for brown for silver nanoparticles, whereas the organic phase remained mostly transparent. So there will be formation of two phases. One is your aqueous phase, then above this it will be organic phase. So two phases will be there, and in the organic phase, what is there in the organic phase? Magnetic nanoparticles are there in the organic phase, so it will come immediately at the interface. The magnetic nanoparticles will be reaching at the interface, and it will lead to the extraction of gold or silver nanoparticles. But it can take some time. It will not happen immediately. In some cases, it can happen immediately, and in some cases, it requires time. Even for few days are required. The system was left undisturbed for several hours, during which the aqueous phase gradually turned transparent, indicating the extraction of metallic nanoparticles by magnetic nanoparticles. Depending on the extraction efficiency of magnetic nanoparticles, the aqueous layer turned transparent immediately or took a few hours. The Gemini surfactant coated surface magnetic nanoparticles undergo strong interaction with SDS stabilized metal nanoparticles and extract them at the interface. So the extraction can take place immediately, or it can take a few hours. Next. So this diagram shows how it is extracted. So these are the Gemini surfactants. So this is the nanoparticle coated with Gemini surfactant. This is the self-assembled Gemini surfactant stabilized nanoparticles. Now in the beaker, this beaker is showing the aqueous layer, and above this aqueous layer will be a small organic layer, and then there will be air, right? Now at the interface, there will be magnetic nanoparticles, and in the bulk, in the aqueous phase, it will be gold or silver. So these will be extracted with the help of these magnetic nanoparticles. So interface behavior in part B, Gemini coated and SDS coated nanoparticles are depicted at the air water interface and in the aqueous phase. Coated nanoparticles, SDS coated nanoparticles remain in the aqueous phase, while Gemini coated nanoparticles migrate to the interface. Now in the final state, that is shown in part C. It illustrates the stabilization of nanoparticles at the air-water interface at the migration of Gemini-coated nanoparticles forming a stable layer. So in conclusion, the modification of iron oxide nanoparticles with surfactants like Gemini surfactant enhances their dispersion stability and interaction with metal nanopollutants, increasing removal efficiency. Surface active iron oxide nanoparticles demonstrate high efficiency in absorbing and removing metal nanopollutants from aqueous solution due to their large surface area and reactive surface properties. Iron oxide nanoparticles can be engineered for selective adsorption of specific metal nanopollutants, making them suitable for targeted water purification applications. The use of iron oxide nanoparticles for pollutant removal is an eco-friendly and non-toxic alternate to the conventional methods aligned with the green chemistry principles. Thank you very much. And now we shall proceed to the lab for the experiment.
ये सारे बारह इको टाइम पे इसकी लाइनेशन
इनका स्टार्ट करो और इसको ना डुप्लीकेट करते हैं। संगीत सिल्वर नैनोपार्टिकल
ਐਰੋਸਲਫੇਟ ਦੇ ਕਾਰਨ ਅਸੀਂ ਇੱਥੇ ਉਹਨੂੰ ਸਭ ਤੋਂ ਜੈਸੇ ਹੀ ਉਹਨੂੰ ਜਦੋਂ 24 ਘੰਟੇ ਲਈ ਹੀਟ ਕਰਦੇ ਹਾਂ ਤਾਂ ਕ੍ਰਿਸਟਲਾਈਜੇਸ਼ਨ ਦਾ ਹੋਣਾ ਠੀਕ ਹੈ ਦੈਟ ਇਜ਼ ਆਵਰ ਤੇ ਅਸੀਂ ਉਹਨੂੰ ਕੈਸ਼ ਬਣਾ ਕੇ ਫਿਰ ਉਹਨੇ ਕੀ ਕਰਦੇ ਹਾਂ ਅਸੀਂ ਉਹਨੂੰ ਹੀਟ ਕਰਦੇ ਹਾਂ ਜਿਹੜੀ ਕ੍ਰਿਸਟਲਾਈਜੇਸ਼ਨ ਉਹ ਕਰਦੇ ਹਾਂ ਸਾਫ ਤੇ ਉਹ ਮੈਟਲ ਸੋਲੂਸ਼ਨ ਵੀ ਬਣ ਜਾਵੇ ਹੋਣੇ ਬਸ ਹੋਣੇ ਪੈ ਜਾਵੇ ਕੌਨਫੀਡੈਂਸ ਦੀ ਵਰਤੇ ਸੋਲੂਸ਼ਨ ਉਹਨੂੰ ਫਿਰ ਅਸੀਂ ਐਕਚੁਅਲੀ ਉਹਨੂੰ ਪਿਉਰੀਫਾਈ ਕਰਨਾ ਪੈਣਾ ਹੈ ਇਹਨੂੰ ਐਸਾ ਚੀਜ਼ ਹੋਣੇ ਕਰਕੇ ਬਹੁਤ ਕਰਕੇ ਕ੍ਰਿਸਟਲਾਈਜ਼ ਕਰਨ ਦੀ ਲੋੜ ਨਹੀਂ ਅਸੀਂ ਉਹਨੂੰ ਵਾਟਰ ਨਾਲ ਪਿਉਰੀਫਾਈ ਕਰਕੇ ਤੇ ਉਹਨੇ ਪਾ ਦਿੱਤਾ ਜੋ ਤੱਕ ਐਨੇ ਵਾਰ ਹੋਰ ਚੀਜ਼ ਕਰਦਾ ਉਹ ਦੇਖਾ Okay, so we are going ahead with this experiment, what we will be doing. So we will prepare the fresh surface active organic nanoparticle. The reason is because we have previously prepared and they are partially working, though they work, but they are not working instantaneously by extracting the gold nanoparticle and turning the solution colorless. So therefore what we will be doing, so we will prepare the iron oxide nanoparticle at the room temperature. The only difference is the reaction will always perform at room temperature, Only thing what we do after the reaction, we place the contents into the Teflon container for the crystallization. So what we will be doing, we will simply avoid the crystallization because that is time consuming, and we will prepare the magnetic nanoparticle the way we have already prepared, and then we will perform the extraction. So she is now preparing the solution, and we will prepare the magnetic nanoparticle.
So you know the procedure to prepare the magnetic nanoparticles. What is to be done? 4 mm of and 8 mm of surfactant. You prepared it yesterday. So we will be repeating this without the hydrothermal process, as explained by Dr. Bakshi.
And we will be using the same sample of gold and silver nanoparticle which you prepared, I think, 2 days ago. Yeah. Okay. So same sample which you prepared. So we'll be using them to see the extraction.
First, they can track the previous sample with this change of intensity of people in the meantime they will run the UV and simultaneously use it. You can take the already prepared gold nanoparticles and already prepared iron oxide nanoparticle. You can check of course there is a change in intensity of the color, but not completely invisible or you can say the gold is still there. But yes, you can check the concentration through the UV. So you can run the UV of the pure sample of the gold, this one, you add the nanoparticles, the iron oxide in this and check the change in intensity, that what is the percentage of absorption, right, or how much amount of the silver is extracted from the sample. So now in the meantime you will prepare the freshly prepared iron oxide and you will repeat this one, right? Okay,
But yeah, one thing that it is better to use the thin test tube and take only 3 ml, not more than 3 ml this gold or silver nanoparticle suspension. Just take 3 ml because what is the volume of the cubit for the UV? I think it is 3 ml.
3 ml. So we can take 1 ml of this and then iron oxide solution will also be required.
No, but 1 ml actually the problem is iron oxide when you are going to add. So 1 ml is insufficient for the filter, right? So you need at least more than 2 ml. Yeah. So what you could do, it's better. Okay, 2.5 ml. So 2.5 ml take the solution, not in this thin test tube, right? So that you can clearly see thin test tube, take one thin test tube, get them the thin test tube, and add 2.5 ml of gold nanoparticle suspension, and how much 1 ml of this. So, and then you will be adding the iron oxide nanoparticle suspension, and just, you don't have to shake, just wait for some time to see if the color changes. Right? So, okay, take this kind of test. So how much, what is the volume of this one, 10 ml?
Yes.
Okay, so 3 ml is something over here, right, up to here, and then you'll be adding that. What is this? ਤੁਸੀਂ ਲਓ ਨਾ test tube. You should take your test tube and go to your seats, please. Please take, two test tubes, one for the gold nanoparticle, one for the silver nanoparticle, and go to your seats. And add 2.5 ml of gold nanoparticle suspension and silver nanoparticles. Keep small beaker and for placing the एक small beaker can place Yeah, you should take one beaker. Everybody should have beakers. Please keep your test tube. Second one also in the same beaker. Yeah. Yeah. And you should have add
Numerical actually, so we have to take the marks in this area. So we will see exactly how much extraction is taken. You do two sample, blank sample, and then this. Is there any change in product?
Can you see this? Side can easily move, and the top layer is of zinc. You can remove the top zinc layer, get off the top layer, and the lower layer can be used for the UV spectrum. So then you can change the color, because this is only the aqueous layer, right? We have added the iron oxide nanoparticle which are in mixing, so there is no change in dilution. Right? If you add water or something which is miscible with water, then you can say, okay, the color may be changed because of dilution, but here there is no dilution because the organic layer is separate and aqueous layer is already. So the color change is only because the gold particles, they are absorbed on the surface of the iron. Right? So this is purely the absorption phenomena, not the dilution or something else. ये magnet लगाओ, remove this hexane layer and you can put it in another tube. So iron will be separated out completely. When we will add it in the cuvette, then we will remove the droplets. You can remove it with the cutter.
those are good action
Pure 15 or 16. 5, 6. ये स्टेबल है, फी में कोई चेंज नहीं आया, फी कोई नहीं आया। क्यूबिट में डाल के पहले पिपेट से
मैं क्यूबिट में डाल के कर देता हूँ क्यूबिट में डाल
के पिपेट से रिमूव करो। इधर सिंक के पास आ जाओ।
absorbance है 0.3 ओके
तुम्हारा एक्सेम वगैरह पूरा निकला नहीं है क्योंकि एक्सेम वो एब्जॉर्ब कर रहा है ना, तो क्लियर सोल्यूशन जो तुम्हारा ब्लैक लगा है
वो क्यूबिट पूरी फिट नहीं हुई ना
हाँ क्यूबिट कम रहेगा।
मिक्स कर दो
absorbance की वाइज नैनोपार्टिकल्स की अब अगर हम नैनोपार्टिकल्स रिमूव कर रहे हैं, ठीक है, तो एब्जॉर्बेंस तो कम हो गया ना, क्योंकि क्लियर सॉल्यूशन है, तो ये वैल्यू से नीचे आनी चाहिए जिसमें
पार्टिकल तो बने हुए हैं, तो शेयरिंग क्रिटिलाइजेशन नहीं हो पाएगा।
गोल्ड वाला है ना, प्योर गोल्ड है। पिछली बार कम दिखा था, हाँ सर वही मतलब गड़बड़ कीबोर्ड वाली फिर लेकिन पहले कहा कि किस प्राइस थी रिमूव करने के बाद आए, ठीक है ना, क्यूबिट खाली याद कर लेना ओवर ले
उसमें कितनी एब्जॉर्बेंस होगी बैक पर भी
5
कोई नहीं, वो एक्सेल फाइल शेयर हो जाएगी।
वेट में ये वाला
फाइल चेक कर लो, उसमें कंपेयर करने की बात है ना। वो तो आपकी आ रही है देखो, ये 400 के आसपास ही है, ये 395 हो गया है ना। तो उस टाइम पे 395 है, 397 आ रही है, तो 395 के अराउंड है। और कितनी है आपकी 1.9 से ऊपर है, ठीक है। इसे सेव कर लेंगे,
नोट कराओ,
बाद में फाइल देखिए।
इसमें कितनी है इसमें जो अटैचमेंट्स आ रही है?
सर 0.48 से नीचे ही है सर।
इसमें पहले कितनी थी?
सर पहले थी 1.92 से नीचे।
अच्छा है इसमें रिडक्शन हो रही है?
थोड़ा टाइम लगा।
अच्छा ये किसकी है? सिल्वर की, गोल्ड की?
गोल्ड की होगी।
अच्छा वो पहले कर अच्छा चलो इनको नोट कराओ फिर पहले फाइल को भेज देना तो नोट भी कराओ।
बेल का इसका बेल नहीं करना, बेल नहीं करना मतलब इसका बिना रिमूव हो गया।
क्या प्लास्टिक
लेयर जो है हेक्सेन में डिजॉल्व होनी चाहिए।
फिल्टर करके फिल्टर
अरे
तो बोल तो ढंग से
पार्टिकल
चेंज वाला डिफरेंस तो आया ना, डिफरेंस आया ना इसमें? ठीक है, इसे ना आप एक काम करो, दो ग्रुप ना एक साथ मर्ज कर दो, ठीक है, और फिर ना एक ही में करवा देंगे क्योंकि क्यूबिट फुल फिल नहीं होती ना तो आता नहीं है सही वगैरह, है ना, कर दो, ठीक है। बाकी ना इनके जो करवाया ना उसकी एक्सेल फाइल आपको दे दी है, ठीक। थोड़ा और फिल करके लाओ, मैं थोड़ा सा मैंने बोला था 2 ग्रुप का मर्ज कर दो पी की जगह, ठीक है।
Most of the movie are trying to win. 11:30 से शुरू होगा।
You should note the reduction in the concentration. Absorbance, it is in the terms of absorbance. Absorbance is proportional to concentration. Okay, so we will measure it in the form of absorbance. Okay, please note down the absorbance.
सुबह तो आया था।
Good afternoon, everybody. We are going ahead with the last one, application of iron oxide in removing metal nanopollutants from contaminated industrial effluents. Okay, yeah. So, in this lecture, I'm going to compile everything that what we have learned. There are two kinds of magnetic nanoparticle we synthesized. The one is the water soluble, other one is the surface active. So we will try to see that how the surface active magnetic nanoparticles, apart from the biological applications, they can be used for certain applications, other applications, environmental related applications. Likewise, how the water soluble magnetic nanoparticles can be used. So what we learned, this is the summarization of hydrothermal synthesis. By using the hydrothermal synthesis, you can synthesize both kind of magnetic nanoparticles, water soluble or the surface active. The only difference is the stabilizing agent or the colloidal stabilizer. The colloidal stabilizer is going to determine if the magnetic nanoparticles are surface active or water soluble. When you are using typically Gemini surfactant, many of the Gemini surfactants, so they lead to the surface active magnetic nanoparticle synthesis, whereas any other amphiphilic molecule which you are using, provided that should be an amphiphilic molecule that has the ability to absorb on the surface of the magnetic nanoparticles, that will lead to the water soluble magnetic nanoparticles. Now we know the characteristic properties, we know the crystal structure, we know the unit cell, everything we know. And now we can see that under what circumstances we can use them for different kind of applications. So predominantly, suppose if you ask me, though I have shown you several images of the magnetic nanoparticle, predominantly this kind of synthesis where I have specifically mentioned in the lab experiment, Dr. Rajiv specifically mentioned that what should be the concentration ratio of ferrous sulfate, ferric sulfate, and that of the ammonium hydroxide along with the stabilizing agent. If you are choosing to keep that ratio close to that ratio, more or less you will be having this kind of nanoparticles. So shape and structure, I would say size particularly, the size will remain this. But if you are going to vary that ratio, you may get different size and different shapes. Apart from that, shape and size also depend on the concentration of the colloidal molecule, that is the surface amphiphilic molecules. So if you are going to vary the concentration of the amphiphilic molecule, now you have learned that in your synthesis in the lab experiment, you use 250 mg of a Gemini surfactant in order to synthesize the surface active magnetic nanoparticle. So because we have standardized the condition accordingly to get the smallest possible size. This is the smallest possible size we are using. Smaller the size, higher is the surface area, and better is the applicability. That is the main purpose or objective for us to produce the nanoparticle with the smaller sizes. And that is why most of the samples, they lead to this much size almost in all the cases. Uh, as I mentioned you in the previous lecture, when we were extracting the blood cells, so I mentioned at that time that we prepared 8 samples of amino acid stabilized magnetic nanoparticles, and then we had approximately 4 to 5 samples of sugar derivatized magnetic nanoparticle. So they are the functional magnetic nanoparticles. When you are going to, if somebody asks you, what is the meaning of the functional magnetic nanoparticle? So functionalization is to put certain kind of functional group on the surface of the magnetic nanoparticle. That could be amino acid, that could be a colloidal amphiphilic molecule, or that could be any polymer. So that is the functionalization. I mean to say that if you do not functionalize this, so then in fact you have the bare surface of the nanoparticle, and the bare surface of the nanoparticle may not be capable of carrying out a precise applicability for a particular kind of application. So therefore, if you want to have uh, 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 if you want to use magnetic nanoparticle for a specific kind of application, then obviously the magnetic nanoparticle has to be functionalized with a specific kind of functional group that would help to facilitate the application. Now, uh, you might be remembering that we use the polyethylene oxide, polypropylene oxide, and polyethylene oxide block copolymers for the templating effect, as I mentioned in the first lecture today. So these polymer, they have the hydroxyl group. When they are going to adsorb on the surface of the polymer is going to adsorb on the surface of the on the magnetic nanoparticle, then these functional groups are available. So now you can use these functional groups for performing certain kind of applicability. This is the meaning of the functionalization. So I'll show you that how we can use such kind of a molecule for environmental applications, and among them the extraction of industrial dyes. You know, you are using dyes everywhere, so you need to change the color of your clothes, so you are taking them to a person who is dyeing it, changing the color, and then everything goes to the drain because dyes are water soluble, most of them, and because they are the organic molecules, so therefore they are the toxic molecules, and our freshwater resources are going to pollute. Dyes are highly toxic and polluted. Dyes are highly toxic and one of the most pollutant of our natural resources on the planet. It is not limited to this country, but everywhere you go, dyes are highly pollutants, and therefore, if you search the publication related to the cleaning of the dyes or cleaning of the contaminated water with dyes, you will see series of publications. So many papers have been published for the cleaning of contaminated water with dye molecules. You won't be able to imagine. So everybody, means this is a very hot area, and what we have done, we have devised certain method on the basis of the magnetic nanoparticle to to clean the contaminated water that is contaminated with a dye molecule. So I'm going to show you that one, and that is shown here in this flow diagram. So these are different samples of the dye solution. This one is highly dark purple dye, blue one, and you may say that this is close to the pink dye. And this is, you may assume that all these solutions are the contaminated water. We want to clean them, right? How we are going to clean them? We have two methodologies. I'm going to show you two methodologies, and by using the magnetic nanoparticle, and how can we clean them. Here we have used water-soluble magnetic nanoparticle, polymer functionalized magnetic nanoparticle. What we do, we synthesize these magnetic nanoparticle, exactly the procedure which you have done in your lab experiment. So then you got this kind of solution when you completed the experiment, and applying with the magnet, you extract the magnetic nanoparticle and you purify it. Eventually, you have purified magnetic nanoparticle. What you can do, you take the dye solution that is contaminated water, you put it into here. Here you have the magnetic nanoparticle. You keep them as you could see over here. So I have taken this one. You may say that suppose we are picking this solution, we transfer this solution into the container which contains the pure magnetic nanoparticle. Functionalize this. You have to keep in your mind that these magnetic nanoparticles are functionalized with OH groups because of the block polymer that is adsorbed on the surface of the magnetic nanoparticle. What happens then? If you keep it some time, eventually you see the dye color is going to be faded away. As more and more dye is going to be absorbed by the magnetic nanoparticle, the dye color is going to vanish. In some cases, entirely the whole dye is completely absorbed on the magnetic nanoparticle, leaving behind a completely transparent solution. In some cases, you may not have a complete extraction, and therefore you may have light color left. It means that some amount of the dye is there in contaminated water. So anything can happen. I mean to say that if you really want to clean the entire solution, obviously you need to have the higher amount of the magnetic nanoparticle. If you take the greater concentration, obviously eventually a point will come when you can remove the entire dye molecules and the solution is colorless. What you can do thereafter, you need to quantify that this much amount of the sample of magnetic nanoparticle is capable of extracting how much amount of the dye molecules. This is the quantification. If you have to tell somebody that I have a new methodology to clean the contaminated dye molecule, then a person will ask you, okay, you tell me how many grams you are taking the magnetic nanoparticle, putting into the dye solution, and how many grams of the dye has been extracted. That is the meaning of quantification. So once I have this dye-loaded magnetic nanoparticles, I can quantify with the different elemental analysis, transmission electron microscopy, scanning electron microscopy, it is XPS, X-ray photon electron spectroscopy. So all these studies can help me to see where the dye molecules have been adsorbed on the surface. They are associated with the hydroxyl group or on the surface. In what form? What is the conformation or what is the molecular status? Is the dye molecule remains intact or it is decomposed? And all those things I can analyze by going through these analysis. Apart from that, I can calculate how much amount of the dye has been extracted by taking how much amount of the magnetic nanoparticle. And suppose you may ask me, how long you have to keep it in the dye solution? Uh, We have performed the experiment and we have seen that some of the dyes are extracted within just few minutes, and the solution you are going to get is transparent, but some of the dyes take a very prolonged time. It means that overnight or maybe 3 to 4 days, eventually when you get the clear solution. So it means that the extraction of the dye depends on the dye molecule, structure configuration of the molecule, and how it is extracted by the functionalized magnetic nanoparticle. So therefore, it is time consuming or a rapid reaction. It altogether depends on the molecular interactions between, between the functional groups and the dye molecule. If the molecular interactions are stronger, as I mentioned in my previous lectures, so then the extraction is instantaneous and rapid. But if the molecular interactions are weaker, then it's a time consuming. So dye molecules are taking time in order to upload on the magnetic nanoparticle. So this is the first method. We call it static method. It means that we have added the magnetic nanoparticle and we observe only. It's a very simple experiment, and we observe that the extraction is happening. There's a second experiment over here. This is very important. The second experiment is the titration experiment. So exactly the same kind of titration which you perform. So in this experiment, what we do, we take the magnetic nanoparticle suspension, purified, we take the purified magnetic nanoparticle and we add pure water and we have the only pure magnetic nanoparticle suspension. We take a burette and in the burette we add the dye solution, right? So we keep adding the dye solution. After each addition, we place the magnet and extract the, collect the nanoparticle and measure UV. After another addition, we, we, we perform the same thing. It means that what we are doing, so we have a burette and we have a beaker. In the beaker, we have magnetic nanoparticle. So what we are doing, we add 1 ml of dye, we shake it, and then we apply the magnet. All the magnetic nanoparticle are taken aside by the magnet, and then we pull the solution, and we measure the UV. Again, we put back the solution. Again, we add 1 that is 1 ml of the dye solution. We perform the same thing. Eventually, a point comes. Eventually, a point comes. Yeah. When we keep on adding, when we add the first 1 ml, suppose we add 1 ml of this solution is completely transparent, because the smaller concentration, when we start adding the smaller concentration, the first lot of dye, when it goes into the solution, it is instantaneously absorbed the magnetic nanoparticle. When you collect it, your solution is completely colorless. If your solution is completely colorless, then you won't be having the absorbance of the dye molecules, right? Eventually, you keep on doing it. A point comes at a certain addition, you see the color appearing. The color of the dye starts appearing. This is the end point of the titration, right? So it demonstrates that at this concentration, you cannot add any more amount of the dye. So the magnetic nanoparticle concentration, they are already saturated with the dye. So they have extracted whatsoever the maximum dye they could extract. Now, we need to find out this concentration. What is the meaning of this concentration? Quantitatively, at the end point, suppose I say that I have taken 1 gram of magnetic nanoparticle, 1 gram of MNPs, magnetic nanoparticle. At this point, just a minute, sorry about it, not 1 gram. I should put the concentration units. So I should say that, okay, I have taken 40 millimolar the magnetic nanoparticle. So this is the lot where the total concentration is 40 millimolar, right? And how much dye has been extracted? Because I'm adding dye and this is the total volume, I can calculate the final concentration of the dye. I may say that, okay, this is probably 2 millimolar of dye, right? What I can do now, I can convert into 40 moles of MNPs, particle. Likewise, I can convert the millimolar into moles. And I can convert, because if the volume is constant, I may say 2 moles, okay? And 2 moles of dye. Now, once I have the moles, I can convert into grams. So it is the grams of magnetic nanoparticles divided by grams of dye. This ratio is required for in any kind of industrial application. The person who is running the industry, he or he may not be chemist. He only needs to know that how many grams of magnetic nanoparticle I have to add in a dye solution and how many grams of the dye molecules are going to be extracted. So that is why this ratio is required. And you could see that with the help of this simple titration procedure, you can this very important ratio. Now, you can have this ratio for all the dyes, right? Or if you want to choose some other kind of functionalized magnetic nanoparticle to facilitate the extraction, you can change whatsoever is giving you the best ratio. Best ratio, what is the best ratio? The minimum amount of the magnetic nanoparticle consumed to extract maximum amount of the dye, that is the best ratio. Am I correct? Because you need a magnetic nanoparticle sample that is much more efficient to extract maximum amount of dye. That is the most appropriate sample. So that is our purpose to check that. So here is the experiment which I was mentioning. As I keep on, as I start the addition, initially there is no absorbance. Eventually you see the dark yellow color. The dark yellow color, yeah, it is this one, though there is one more over here, but this addition represent this much concentration. After this, there is a bright green, and eventually this is. It means that end point has come over here. It is a 0.08 millimolar of the brilliant blue dye. So this is brilliant blue dye solution. So this is the concentration. The end point of this titration is somewhere over here. So now you could see that what we have done, we have titrated, we have performed the titrations under different pH conditions. The neutral pH, basic pH, and the acidic pH, just to see that which pH environment is going to give us the maximum extraction. What we have seen over here, you see that. So in the case of the acidic pH, yeah. So you see that in the case of acidic pH, close to 1 millimolar of the dye is being extracted. It means that's very high in comparison to the neutral and the basic. Neutral and basic, you may assume close to 0.1 millimolar, 10 times high. If you are performing this titration under acidic condition, so you can extract dye up to 10 times in comparison to the neutral medium, right? And so we took 6 dyes. It's 1, 2, 3, 4, 5, 6, and we performed the titrations just to find out the which dye is giving the best results. So we found that this is the brilliant blue that is giving the best results. So the extraction is close to 1 millimolar for this one under pH, acidic pH condition. Likewise, this is I forget its name. What is? Excuse me? Yeah, black tea. Sorry about that. Actually, it just went out of my mind. So it is Eroculum black tea, right? And the black tea also performing excellent results. So though it is not equivalent to that of the brilliant blue, but still close to 0.8 millimolar. So in comparison to the required sample of the magnetic nanoparticles, you can extract it. Then we have methyl violet. Methyl violet is not working appropriately. It is working more in the basic condition rather than the acidic condition. You see, the acidic condition is the minimum extraction. Whatsoever we extraction observed, so that is in the basic condition. And surprisingly, all these three dyes, this is methyl orange, uh, rhodamine blue, and this one is bromophenol blue, correct. So all these dyes were not extracted at all under any pH condition, acidic, basic, or neutral. So what is the meaning of the functionalized nanoparticles application? I say that you cannot expect that the functionalized nanoparticle, magnetic nanoparticle which you have synthesized, is going to be is going to work for all kinds of dye. That is not the case, right? It is the functional group which will decide that how it interacts with the functional groups of the dye. Wherever the functional group that is on the magnetic nanoparticle, if it has the stronger interaction, in this case it has a stronger interaction with the brilliant blue and this dye, so because of that the extraction is maximum. But this functional group has minimum interaction, so extraction is not happening with the titration. But if you perform the static extraction, like the static method, so then all dyes are extracted, though they are not equivalent. But even those not shown in the titration method, so they are being extracted. The reason is the titration method is very rapid. You are adding dye, maximum 3 to 4 minutes you are going to wait, and you are going to measure the UV. But in the static method, you will wait for overnight, more than 12 hours or 24 hours. That is not the the uh, 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 the time period you are going to consume for the titration. So titration is a rapid method, and if you want to sell this technology methodology to some industry, industry will prefer the titration method rather than the static method. So they will prefer that we need a kind of a rapid method which can instantaneously extract the dye in a shorter period of time. So bottom line, this is the way we choose, this is the way the functionalized magnetic nanoparticle work. The main purpose to show this is to just tell you that how we can design synthesized functionalized magnetic nanoparticle for a particular application, and on the basis of that application we could see that how much applicability this kind of functional magnetic nanoparticle can have, and in this way we can further modify or improve the method. So how the extraction is happening? The brilliant blue is showing the maximum extraction in the acidic medium rather than the basic medium. Here is the story. In the basic medium, actually, the hydroxyl groups are deprotonated, right? They are negatively charged. The negatively charged group, they are not going to interact with the sulfonate group. So this is the sulfonate group of the brilliant blue. So therefore, they are going to interact with the positively charged nitrogen here. But the problem is this positively charged nitrogen is screened with a bulky group that is screening the electrostatic interaction. Remember I told you, it's not always the interaction, electrostatic interactions is going to work. You have to find out that if there is any bulkiness, screening, or the steric hindrances which are impeding the electrostatic interaction. If that is the case, you may not have a sufficient kind of extraction. On the contrary, you see over here, when you have protonated hydroxyl group in the acidic medium, This is not a problem. So for the protonated hydroxyl groups, so the electrostatic interactions can easily be performed with the terminal group like this sulfonate group, and hence the extraction is very rapid. So I mean to say that these, you need to have the knowledge of, the understanding of every kind of fundamental understanding which is involved in the procedure to understand or to apply it appropriately. So I mean to say that the knowledge of the basic chemistry is very, very important. If you are clear about the basic concept, I don't think there is any issue when you have to perform any kind of research activity. So this is the one, this is the example of the extraction of dye. I just wanted to show you that how functionalized magnetic nanoparticles work while performing certain kind of applications. And this is specifically being used by using the block polymer or block polymer. So these are the water-soluble magnetic nanoparticle. Sorry, yeah. Another thing, suppose so you might be asking me, what is the purpose of transmission electron studies or scanning electron microscope? You see the images. Suppose if you ask me, where are the dye molecules? We can see them. These are the magnetic nanoparticle now carrying the dye molecules. You cannot see them in the black and white. But if you apply the elemental mapping, these colored images are known as elemental mapping. Elemental mapping means if I apply to see only the iron, then I will see the color of the iron. I'll see a colored area. Iron is present in the sample. So this color, you can have any color. I mean to say you can change the color in the instrument, but if you see the colored area, other areas are completely dark. It means that that area belongs to iron, right? So you see that these are the iron oxide nanoparticles. So therefore, iron is there. Oxygen is also there. So it is iron oxide. Now you want to see the carbon. The thing is that you need to know where the carbon is, okay? The carbon is here, methylene group, methyl group, methylene group, they are all carbon. It means the carbon is in the polymer backbone. Apart from that, the carbon is in the dye molecules. So your dye molecules, they have all carbon. It means that this carbon is because of the carbon from the polymer as well as the dye. You cannot differentiate that which carbon is of the dye and which carbon is the polymer. But at least you see the ring structure. Can you see the ring structure? The ring structure shows that the carbon is coated around the nanoparticle. That is why you have a ring structure. Because if it's a roughly polyhedral nanoparticle, it means that if it's coated with the polymer, carbon is originating from there. And if the dye is being absorbed, dye is going to interact with the polymer molecule, so therefore the carbon is there. So since inside the ring structure, the dark areas, because it is more iron oxide, so therefore the emission of the carbon is not that much, so therefore that is not a bright green area, though the carbon is having a layer around the magnetic nanoparticle. What is unique only to the dye? That is nitrogen. Nitrogen is only available in the dye molecules. It's neither the part of iron oxide nor the part of dye. Nor the part of polymer. Polymer is not having nitrogen. No nitrogen functional group in the polymer molecule. And there is no nitrogen in the magnetic nanoparticle. It's iron oxide. So therefore, nitrogen is here, here, and here. So therefore, the nitrogen which you see is the part of the dye molecule. So therefore, in the sample, you see the mapping of the nitrogen. That is only because of the dye molecule. That shows, now you see that shows the extraction of the dye. The nitrogen mapping clearly indicates that it is available on the surface of the die. You see the circle area, you see some of the tiny circle area. So therefore, it is the part of the shell that is created around the magnetic nanoparticle. And apart from that, you have the sulfur as well. Though the amount of sulfur is very less, this is the only functional group, sulfonate, that is the origin of the sulfur mapping. And so therefore, both the nitrogen and the sulfur mapping confirms the presence of the dye. So I mean to say, by applying all results, you come to the mechanism of the extraction. How do we know that the nitrogen or dye is not decomposed, right? If it's not fragmentized, I'll say it looks that the dye molecule is intact. There is no decomposition because we have the nitrogen emission as well as that of the sulfur. So we could map the sulfur as well as the nitrogen, and we can also quantitatively on the elemental basis, we can see how much is the atomic percentage of nitrogen, sulfur, and carbon, and we can see that, and we can calculate the same ratio. The ratio we have calculated over here, the same ratio we can calculate over there from the imaging study. So this is the advantage of imaging study, but you need a very high resolution to get these images, and very highly sophisticated transmission electron microscope. Usually they are not available because they are very, very expensive. So you have to perform the experiment very carefully under uh, entirely non-contaminated, because even a single dust particle would create the elemental emission from the element, which you will be surprised that why this elemental emission is there, and then you will realize that it is because of the contamination. So I mean to say that when you have to work in a very clean environment, and no matter you are working in a nanomaterial, any kind of research, inorganic, organic, or physical chemistry, so you have to have a clean environment and non-contaminated environment to quantify the research. So sustainability, and remember I told you that sustainability in the first few lectures is a very important aspect now. Now we want that we should have whatsoever kind of action we perform in our day-to-day life, we should have certain degree of sustainability. I mean to say simple action is when you are using the white sheets. So we want that the white sheet after the usage, we don't throw in the garbage. They could be recycled to produce the fresh white sheets. The plastic we are using, it should be recycled, and we should have another lot of the fresh plastic which can be used for another purpose. So that is the meaning of sustainability. But it's the same way. So if we could reuse, now you know that when you have to use the reverse osmosis for water purification instrument, you have everybody in your home, right? So you are changing the cylinders. So I don't know if they are reusable or they are regenerating them, how they are doing that. But in this case, you can easily do. How? See over here. So here is the brilliant blue solution at pH 1.5. When I'm going to add magnetic nanoparticle, in pH 1.5 extraction is highly facilitated. Instantaneously, the whole extraction is going to take place and your solution is completely transparent. Okay, and you could see it. Here is the actual solution. And when I apply the magnet after adding the magnetic nanoparticles are over here. You see the transparent solution? Entirely the solution is transparent, right? What I do, I pick the nanoparticle. It means nanoparticle have picked all the dye molecules from this solution. And I picked the nanoparticle, put it in the basic solution. The pH is 12.5. I know already from my results the interaction of the dye and the magnetic nanoparticle least in the basic region. So therefore the dye will be released. The entire dye which was extracted over here is further released into the same bottle because you have changed the pH. What you could do, you again collect the magnetic nanoparticle, purify it. I mean to say that This is the purified water. You have thrown away the dye in some, I would say, the discarded bottle, right? And then again pick the same lot of magnetic nanoparticle, put it in the fresh lot of contaminated water. You can recycle the same lot of magnetic nanoparticles. You don't need any fresh magnetic nanoparticle. And here is the cycle. So we have performed up to 4 cycles, and the concentration of the released dye, that remains more or less same, close to 1.5 millimolar in all the cases, and this is the UV. So for all the 4 cycles, the UV plots are quite close to each other. So I mean to say, this is known as sustainability, right? So what I want to say that in our research, we should now be more careful that how I can have, how I can include the sustainability at some point in my research. By adding the sustainability, it means that you are generating a new field of the research so that your natural resources, our natural resources, are not going to be exhausted. That is the main purpose of the sustainability. Oh, sorry about that. Uh, I wanted to show you some other application by using the cyclodextrin gold model pollutant. Earlier, I showed you the extraction of the model pollutant by using some kind of surfactant, but this was by using the cyclodextrin. But I have to stop over here because the first application, I never realized that it's going to take so long. Anyway, so I should I should I should keep it off, right? And I should finish it over here. So hopefully you feel this is a very interesting application where you can have the sustainability as well. And now I'm directly going to the conclusion part. Surface active magnetic nanoparticles are a new class of nanomaterial with interfacial application. You have already seen the interfacial application. The most important among them was the extraction of the blood cell. That was directly related to the bioapplicability. They remove pollutants without incorporating in aqueous bulk. This is what we saw when we took the gold and silver nanoparticle as model pollutant, and we try to use the surface active in order to extract the metallic pollutant, and those metallic pollutants means the industrial effluents from the metal industry. So how we can clean that contaminated water which is with that of the industrial effluent. They possess enormous potential to use in the biological application at biointerfaces. So I have shown you just one application, that is the extraction of blood cell or the protein film formation, but there is huge range of bioapplicability at the biointerfaces. Among them, usually the magnetic hyperthermia, MRI that you are being means it is a very common. So all these applications particularly require some in-depth analysis of in-depth knowledge of biology. So this is all about the entire course. Hopefully you have understood some part of the course, if not the entire course. And what I believe that should be useful for you some point of at some point in your career, whether you are pursuing research or not. That is not important. But even having the knowledge of material science or chemistry, that is very, very important. So at the end, what I would like to suggest you from my experience that how I became a scientist. I feel still not a good scientist, but still I'm trying at this age. It's a journey, long journey of more than 30 years. I still remember when I was student doing PhD in Punjab University. So it is more than 30 years ago. So at that time, I never thought that I'll be able to publish this much. and I could do this much research. But how it happened, and if you ask me just in one sentence that how it happened, I'll tell you that only the basic chemistry. If you understand the basic chemistry, fundamental chemistry, first year chemistry correctly, quantitatively, there is no problem in the research field that you cannot solve. This is, I'm telling you, from my experience. And the day I started understanding the fundamental chemistry, it, it, it was not the case when I was doing my PhD. I, I had so much confusion. The basic chemistry was not clear to me. The fundamental chemistry at that time was not clear to me. But eventually, when I started teaching and slowly reading textbook and everything, so the most advantaged, I would say the most important time obviously was when I was in North America. We have excellent textbook for the introductory chemistry courses, and with clarity of understanding the concept, applying quantitatively, right? So that helped me a lot. So it is not that easy to buy those textbooks because they are very expensive. But I would suggest you that you can tell your library or to your department at least one or two copies of such kind of recent textbooks which are being used in good universities in North America or in United States. So they are not that expensive if you have to buy only two copies to keep in the library. So one copy costs usually our 10 textbook costs, which are required by the every student. Every student has to buy it. It's $150 close to, and sometimes they reach to $200 per copy. So there it is required, and every student is required to have that before they enter into the course or come to the first class. So here we cannot impose such expensive textbook, but I would say that that is very important thing. And eventually, though textbook really help, but if you do not have an interest in chemistry, then textbook cannot help you, right? So therefore, you have to create your interest in chemistry, the fundamental chemistry, to understand it, and eventually try to apply it. So at the end of the course, so I'm so grateful for you, all of you, who spared your valuable time to go through this course and try to understand the aspects which we have included. And hopefully, they will be useful for you in your future career. Thank you so much. Any question, please, if you have.
endeavors. We deeply appreciate the time and efforts you invested in sharing your knowledge and engaging in meaningful discussions. Your presence has truly made this event a memorable and impactful experience. Thank you once again for your support and dedication to the advancement of knowledge.
I now invite Dr. Jaspreet Kaur Rajput, course coordinator of the GIAN program, to share brief reports of the entire course.
A very good afternoon to everyone. First of all, I would like to extend my warm welcome to the dignitaries, Dr. Mandeep Singh Bakshi, Dr. Rajiv Jindal, Professor N.C. Kotial, and other faculty members of the department, students, participants. So as you know, this GIAN program is now completing today. So this is the 5th day of our GIAN program, and during these 5 days, the participants, they were enriched with the surface chemistry and different surface active nanoparticles and the applications. So if I'm talking about the participants of this course, we were having a number of students from different diverse background. We were having students from engineering, from basic sciences, research scholars, faculty members, and even graduation and post-graduation students. So 46 students have registered in this course, and out of these 46, 41 students have physically attended our course. So in these 5 days, total 12 lectures were delivered and 5 tutorials in which we were having hands-on session and lab experiments were there. So I'm giving you a brief report of this course. So on day 1, we delved into the quantification of the solute-solvent interactions and explored the microheterogeneous aqueous phase for nanomaterial synthesis. So in that tutorial, there was a hand-on experiment on the freezing point depression and boiling point elevation, which provide a solid foundation for understanding these fundamental concepts. On day 2, we focused on the fundamentals of surface active species and quantification of bulk and interfacial processes. The tutorial involved synthesis, purification, and characterization of Gemini surfactant 12-12. So this gives participant practical experience in the synthesis of the Gemini surfactant. On day 3rd, we covered the quantification of the crystalline solid and biointerface material chemistry. In the tutorial, the participants synthesized, characterized, and studied the electronic properties of gold and silver nanoparticles, gaining valuable insight into their properties and applications. On day 4, it was dedicated to the bulk, soluble, and surface active nanomaterials for biointerfaces, focuses on surface active iron oxide nanoparticles. So in the tutorial, there was a synthesis of Gemini surfactant stabilized surface active iron oxide nanoparticles, and today we explored the understanding of nano applicability and applications of iron oxide nanoparticles in blood cell extraction and removing the metal nanopollutants from contaminated industrial effluent. The tutorial was focused on application of surface active iron oxide particle, which were synthesized on day 4 for the removal of metal nanopollutants from the aqueous bulk. So I feel that the overall course was very enriching and participants gained lots of knowledge in this particular field, and I thank Dr. Bakshi for accepting our invitation to be here with us, and thank you so much, sir, that you have enlightened our participants. I hope in future also you will collaborate us in these academic activities. Thanks a lot, sir.
Thank you, ma'am. Now we have the privilege of hearing from our esteemed guest. It is an honor to invite Dr. Mandeep Singh Bakshi to share his experience at NIT Jalandhar.
Good afternoon, everybody. This was excellent experience and remarkably excellent. The reason was it is first time I have seen the students were so attentive, very serious in attending all the lectures. And I'm really thankful to you all because I think right from the first day I mentioned that if you really want to learn something, and then please do not skip any of the lecture. And I'm so happy that everybody whom I saw on the first day, and he or she is still there today. So it was really a great success. I am so happy and thankful to Government of India for providing so much money for this kind of academic activities, and hopefully this will be really helpful for you in your future career advantages. So thank you so much, and I also thank to Dr. Rajput, Dr. Rajiv, and the faculty here from the Department of Chemistry in NIT, as well as director of the NIT, and everybody who helped us to achieve the goals or the objectives of this course. Thank you so much.
Thank you, sir, for your inspiring word and encouragement. Now it's time to recognize the efforts of all of our participants. We will begin the certificate distribution ceremony. I request Dr. Mandeep Singh Bakshi, Dr. Rajiv Jindal, Dr. Jaspreet Kaur Rajput, Dr. N.C. Kothiyal, and Dr. Balbir Singh Kaith to kindly do the honors. Participants, please come forward as we call out your name.
Before the certificate distribution, I will request Dr. Kothiyal to say a few words.
अच्छा इसका बोल रहा था नहीं मैंने तो ये बोला था कि
Honorable Professor Bakshi, resource person of this program, Professor B.S. Kath, Head Department of ITEP, Dr. Jindal, Head of the Department and the course coordinator of this program, Dr. Jaspreet Kaur, the course coordinator of this program. I know that both of them are working very hard for past many days. Such kind of programs requires a very hard work. You might not be knowing that how much homework has gone behind the scene. And since I was associated with the GIAN program at NIT Jalandhar since 2016, and among the first programs, I think me and Dr. Kath has organized 2 programs in chemistry here, beside the other programs which were organized by the various engineering departments. And since then I know that the main purpose was to expose you with the foreign faculty. It is not possible for you to go foreign individually. So if the expert is coming to your doors, you can gain a lot of knowledge in the concerned area. And that was the purpose. If it is solved now, you can interact with the expert in times to come, and that will give you boost up. I think in the research area, whenever you are stuck in the field of maybe in nanotechnology, or I say in general it is surface chemistry. Surface chemistry is such a wonderful subject, I think, which you can find use in almost all areas. Whether you are engineer, you are biologist, you are chemist, or you are anywhere in the world, you will find that surface chemistry will always come there somehow or in some place. So it's a very interesting area. Keep on growing in this area, and I wish all good success for you all in future endeavors. And I hope that you must have gained a lot of knowledge out of this course. Thank you for coming to this place. You are from different places. And thanks to Professor Bakshi, who has come all the way from US and devoted a long time and given a lot of energy for conducting such kind of courses where continuous lectures has to be delivered. Thank you very much.
Thank you, sir. Now I request Dr. B.S. Kath to please come and share the experience.
Good afternoon, everybody, and namaskar. Honorable Professor N.C. Kothiyal, Professor HAG and Senior Most Professor of Chemistry Department, Dr. M.S. Bakshi from Wisconsin University, US, and the expert speaker of this course, 5-day course. Dr. Rajiv Jindal, Head Department of Chemistry and coordinator of this program, this GIAN program. Dr. Rajput, other course coordinator of this program, my worthy other faculty members, dear participants, it's really a great honor that Dr. M.S. Bakshi is with us, who has come all the way to NIT Jalandhar. And before I go ahead with this, I think I must congratulate both of 40 or 50. Yeah, only 9 programs we got in at NIT Jalandhar, and Department of Chemistry is one of them. So we must be proud of Dr. Jindal and Dr. Rajput. So बहुत अच्छा इसका मतलब प्रोग्राम में काफ़ी कंटेंट है, और कंटेंट आई हैव सीन हियर डेफिनेटली द प्रोग्राम इज ऑन iron oxide nanoparticles for biomedical applications, and I'm sure you must have learned a lot about the iron oxide, how important it is for the living organisms, हीम आयरन एंड नॉन हीम आयरन आपने सीखा होगा इसमें काफ़ी, और सरफेस केमिस्ट्री और उसका फंडामेंटल तो काफ़ी आपने लर्न किया होगा इसमें, क्योंकि डॉक्टर बख्शी इज द मास्टर इन दैट फील्ड, इज रियली मास्टर इन दैट फील्ड, वी नो दैट. और इन्होंने मुझे जो पता चला, क्योंकि मैं फीडबैक की अपने स्टूडेंट्स की, मेरे स्टूडेंट यहाँ बैठी है तो उनसे मैं फीडबैक डेली की डेली लेता था, तो शी टोल्ड मी कि पूरा हमें फंडामेंटल्स बताए जा रहे हैं, बेसिक्स कंप्लीट, तो दैट्स अ गुड थिंग. आई एम श्योर एवरीवन of you have learned a lot about the surface chemistry, about the nanotechnology, about the nanoscience, what is nanoscience, और nanoparticle बनाए कैसे जाते हैं, and what is the importance of such materials in living organisms, for especially for drug delivery. So क्योंकि iron oxides they play very important role. तो आपने काफ़ी कुछ सीख लिया, मुझे बताने की ज़रूरत नहीं है. तो वी आर हाईली थैंकफुल टू डॉक्टर एम.एस. बख्शी, जो इतनी दूर से आए और इतना कुछ हमारे स्टूडेंट को दे गए. एंड आई थिंक एवरीवन ऑफ यू मस्ट हैव द कॉन्टैक्ट नंबर ऑफ डॉक्टर बख्शी. ले लिया सबने नंबर इनका? कॉन्टैक्ट नंबर नहीं दिया सबने? ईमेल आईडी तो ले लो. है सर.
So anybody can contact me by going through the Department of Chemistry website on University's website. Yeah. So you are welcome. Do not hesitate to contact me anytime. You can contact me.
सर डायरेक्ट ईमेल आईडी दे तो ज़्यादा ठीक रहता है. इफ यू डोंट माइंड, वो यार ठीक रहता है. नहीं, बच्चे बेचारे कहाँ ढूंढते फिरेंगे. यस सर, दैट वुड बी बेटर सर. आप देखो सर, बच्चे कितने खुश हो गए सर. सर दे आर इनोसेंट सोल्स. सर इन्हीं में भगवान बसता है. ये भगवान रूप यही है सर. Now you can note down.
You are now that students, you can contact him anytime.
No, सर का कॉन्टैक्ट नंबर मिल गया. देखो आप व्हाट्सएप पे भी आप बात कर सकते हैं सर से. नहीं, ग्रेट सर, थैंक यू सो मच सर. almost 12 hours difference. Almost 12 hour difference है. So thank you so much sir. बहुत अच्छा. तो यही चीजें जो मैटर करती थ्रू आउट लाइफ, और आप लोगों ने बहुत कुछ एक्सपोजर लिया है, आई नो. तो दैट इज द डिफरेंस कि जो बाहर से कोई भी आता है उनसे बहुत कुछ सीखने को मिलता है. आई एम श्योर यू मस्ट हैव लर्नड अ लॉट आउट ऑफ दिस प्रोग्राम. तो once again thank you sir, and congratulations to both the coordinators Dr. Rajput and Dr. Jindal, and thank you so much. Jai Hind, Jai Bharat.
Thank you, sir. Now I request Dr. Mandeep Singh Bakshi, Dr. Rajiv Jindal, Dr. Jasprit Kaur Rajput, Dr. N.C. Kothiyal, and Dr. B.S. Kath to kindly do the honors. Participants, please come forward as we call out your name.
Our first participant is Dr. Prabjot Kaur. Next, Dr. Kuljit Kaur. Next, Dr. Kuljit Kaur. Next, Mr. Arvind Singh. Mr. Dinesh Verma. Mr. Nitin Kumar. Miss Samriti. Miss Sareen. Miss Avjot Kaur. Miss Garima Sharma. Miss Komal Nandal. Miss Princepreet Kaur. Mr. Rahul. Mrs. Jyoti. Miss Bindu. Mr. Rohitash Kumar.
Miss Jyoti Panu. Miss Sikha Sharma,
Mr. Gagnesh, Miss Anushka, Mrs. Shikha, Miss Mehak Sharma, Miss Himani Sharma, Miss Mahashweta, Mr. Siddharth, Mr. J. Sharath, Miss Deepika, Miss Priti, Miss Ishrat, Miss Mansi, Miss Jasprit Kaur, Miss Kritika Sharma, Miss Pragya Verma, Miss Arushi, Miss Ananya, Miss Payal, Miss Prabh Simran, Miss Jasneet Kaur, Miss Jasneet Kaur, Mr. Pankaj, Miss Amanpreet, Miss Jyoti Rani, Mr. Saurav Singh,
Now I invite the organizing team to come forward and collect their certificates as a token of appreciation for their hard work and dedication.
First, we have Dr. Meenu Singla, Miss Arpita, Miss Samriti Dogra, Miss Sareen, Miss Avjot Kaur, Miss Komal Nandal, Miss Princepreet Kaur,
I really want to thank our organizing committee, especially the research scholars. Without these research scholars, this program could not be possible. So thanks a lot and big rounds of applause.
Mr. Rahul, Miss Jyoti, Miss Bindu, Mr. Rohitash Kumar,
Miss Jyoti Panu, Miss Shikha Sharma, Mr. Gagnesh, Miss Anushka, Miss Jyoti Rani, Mrs. Himani Sharma, sorry. Miss Himani, Miss Anokhi,
Mr. Janak, Mrs. Jyoti, Mr. Abhinav Gupta, Mr. Rishabh, Mr. Rajat, Miss Shreya, Miss Navpreet Kaur, Mr. Arvind, Congratulations to all the participants for successfully completing the program. Now I invite Dr. Rajiv Jindal, course coordinator, for the vote of thanks. Please, sir.
So, good evening to all of you. Respected Professor Mandeep Singh Bakshi, Professor N.C. Kothiyal, Professor B.S. Kait, Dr. Jaspreet Kaur Rajput, Dr. Vimal, Dr. Vikram, Dr. Rakesh, Dr. Sadhika, Dr. Sanjeev Sutradhar, Dr. Meenu, and dear participants, as we have gathered here to mark the conclusion of GIAN program on iron oxide nanoparticles for biomedical applications, fundamentals of bioapplicability, I am filled with a sense of pride, gratitude, and accomplishment. On the behalf of Department of Chemistry and our institution, I would like to extend my heartfelt thanks to each and every one of you for making this program a resounding success. First and foremost, I would like to thank our distinguished speaker, Dr. Mandeep Singh Bakshi and Dr. Jaspreet Kaur Rajput for sharing their expertise and insights with us. Your lectures and interactions have been truly enriching and inspiring. A special thank to Director Professor B.K. Kanodia, whose guidance and leadership have been instrumental in success of this initiative. I would also like to thank our participants who have traveled from far and wide to join us. Your enthusiasm, curiosity, and engagement have been driving force behind this program. To our colleagues and staff members who have worked tirelessly behind the scene to ensure the smooth execution of this program, I offer my sincerest gratitude. Your dedication, commitment, and teamwork have been exemplary. I would also like to thank the Ministry of Education for supporting the initiative through the GIAN program. I would also like to acknowledge the contributions of all the organizing committee members, Komal, Arvind, Smriti, Prince Preet, Jyoti, Sreen, Rahul, Avjot, and many other research scholars who have worked hard with dedication and enthusiasm and have made this program a success. As we conclude this program, I hope that knowledge, ideas, and connections forged here will continue to inspire and motivate us to pursue excellence in our respective fields. Once again, I thank you all for being part of this journey. I wish you all safe and pleasant journey back home. Thank you very much.
Thank you, sir, for acknowledging everyone's efforts and contribution. We come to the end of the GIAN program closing ceremony. On behalf of the organizing team, I thank each one of you for your participation, enthusiasm, and support. And also thanks to the Ministry of Education for initiating such events.
Now we request everyone to stand on their seats for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तब शुभ आशीष मागे गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Students, those who have to appear for the examination tomorrow, the venue is room number 109 at Chemistry Department. Timings are 10 a.m. to 11 a.m.
Thank you and have a wonderful day. Now you are all invited for the high tea and snacks.